Thank you all very much for coming. We've got a great group, and this doesn't include, of course, all the people who are remote and online and will be listening in. My name is Bert Kosiski. I'm president of the Board of Trustees of CureCadasil. I want to acknowledge a couple of other trustee members we have here who will be helping out. Jane Gunther, who is a past president and science officer of CureCadasil and the official photographer of our event. And then Pedro de Lancaster, who is also on the board of trustees and will be looking in the chat and maybe passing microphones around, which reminds me of a couple important announcements. One, everything will be recorded. Okay, so be cognizant of that. And we will post all these recordings on our website, curecadasil.org. The other thing is any comments or questions here in the room should go through one of the microphones, or else people who are online won't hear them and it won't be recorded. So it's very important. And I think that was it. I do want to thank and acknowledge the United Leukodystrophy Foundation for supporting this meeting. This is our second annual one. They also supported the one last year. It was very gracious of them. And we also have funding from the CZI grant. It's a CZI patient-partnered collaboration with neurodegenerative diseases. So those two acknowledgements, I think, are important to make. And then, of course, all of our speakers who you're going to get to hear today. So next slide. Oh, sorry. And in my hand. You know, I should remind everyone the purpose of this whole meeting, I think, is to improve communications between patients and investigators. Okay. So it's a two-way street. You know, we would like investigators to hear firsthand from patients what their concerns and questions and issues are, and vice versa. And last year it ended up being more of a traditional investigators presenting their wares and where they're doing, which was great. They were great presentations. And then patients asking them questions and getting answers. And so we tried to put a little bit more emphasis this year on actually having upfront patient input and directly from what they want to say and how they want to express things. But still maintaining the output from the investigators and where they are with the studies, and then ending up with a grand finale of getting a good discussion going between both groups. So that's our goal. So we'll do this by the first three talks are all coming out of observational studies here in the United States. And a couple of these are shortened. All of these times are a little bit tentative. We aren't going to cut anybody off today. I don't think we'll have plenty of time. Two of these talks were also from last year, and they're updates on where those studies are. And then we'll take a break in here. And then we have two guests from abroad who from Europe who are going to tell us about their actually some very exciting research that's going on both from Leiden and also from Stockholm. And then we'll end, as I said, with a panel discussion, getting everyone together, and we'll figure out how to work that with chairs and whatnot. And then after a brief announcement, I want to point out that there will be an afternoon session of the consortium. The consortium study here in the United States has set this up, and it will be, I think, a very good thing to attend if you can stay around and are still here. This will be at 4 o'clock in this same room, and it'll be followed by a half an hour informal session after that, which will just informally meet and greet type of thing before dinner. So the first thing up is a joint effort here by our patient, and it comes out of our CZI grant, the Patient Partner Collaboration in Neurodegenerative Disorder Studies. And this is an effort of a patient group, largely a community advisory group, excuse me, that has put a lot of effort into thinking of what to do and how to inform the research and community. And it's been headed up also by our PI from the investigative side of the grant, Fanny Elahi. And Fanny is a physician scientist. Whoops. Sorry. Fanny is a physician scientist at Mount Sinai. Her research is focused on understanding the link between disease of the brain's small blood vessels and neurodegeneration in CureCadasil. Her team is modeling disease using blood cells from patients and research participants to make discoveries in therapeutic potential and find a cure for CureCadasil. In 2022, CureCadasil and the Elahi lab at Mount Sinai were awarded, as I mentioned, the Chan Zuckerberg Initiative Patient Partner Collaborations for Rare Neurodegenerative Disease Grant. And shown here is the below and is the whole panel of the group of community advisories, some of which will be here and presenting today. So without further ado, I'll turn it over to Fanny.
Thanks so much, Bert. So it's such a pleasure to be here. This is a highlight for me, the most important conference that I look forward to all year. And I have to say that as a physician scientist, I just feel that I really hit the jackpot. This started as an intellectual work for me. I was fascinated with white matter, and I wanted to understand why white matter is impacted in the brain and why everyone thinks it's a small vessel disease. Being at this conference, we know it's not just small vessel disease. And then as I started thinking about what would be the most impactful thing to do, I realized we need to understand fundamental biology that leads to that. And there's no way to do that outside of monogenic diseases. It was still very intellectual. So I was like, okay, what are the genes that are impacting white matter and, progranulin and Notch3 and COL4A and all the different kinds of genes? And circumstance had it that I started becoming known as the white matter expert in the Bay Area and started being sent patients, and they all had CureCadasil. And once I met patients who are impacted by CureCadasil, I got hooked and it became very personal. So this is very personal for me, as it is for you, and it's very personal for my team. We are now on a mission. That's how we do the research that we do. And this is why we applied for this grant. This grant is very different. Chan Zuckerberg Initiative is an initiative that funds fundamental knowledge, basic fundamental knowledge in science across the board of many different diseases impacting from developmental childhood disorders to degenerative dementia causing diseases. And for the first time, they put an RFA out for a patient partnered research. And around that time, despite being a physician scientist, I had realized that I don't really know what bothers people, actually. I think I know, and I don't really know because we're not ever working with the patients to come up with the outcomes. And in order to do biological discoveries that are going to be impactful, we need to know what we should target. What if it's not the headache or the cognitive impairment that actually matters to the patients? What if it's something totally different? And so 5 grants out of 300 got funded, and we did win the jackpot. And that was thanks to CureCadasil because our relationship had started before that. And I think it was just meant to be. And ever since then, it's been incredible. Getting to know you guys as part of the community advisory board has been amazing. And now when I apply for grants and I don't get it, I hear things like, don't worry, the next one will happen and we're doing this together. So I feel for the first time as a researcher that I'm not isolated in a void, competing for intellectual things, that there's true meaning to what we're doing and that we'll get there. And we may get very lucky like we did for this funding, or it may be a little bit less luck, but we'll get there. And I think the other thing that has happened is that all the different research projects are also sort of coming together. We'd like them to come together a little bit faster, but we are. breaking down the silos and actively thinking about how do we decrease the burden on the research participants and how can we share data and who does what in this. So just amazing. And how another so 2 years of this grant have gone by and it's been incredible. Some papers are already out, more coming. I think we actually have two really exciting biological pathways that we're now thinking about from a therapeutic angle. And all of that just emerged out of nowhere over the past 2 years. At this year's basic science meeting of Chan Zuckerberg Initiative, they are featuring Cadasil as the disease for everyone to hear about. And that speaks, I don't know, lengths to how this community has come together to raise awareness. And this is something that my panelists are going to talk about. We really think that raising awareness is going to achieve great things, because if people hear about the opportunities to push forward knowledge towards therapeutics, everyone is going to bring in whatever they can contribute that would be complementary to what is existing. So without further ado, I want you to hear from the panelists a little bit about who they are, and we're going to feature a concrete project that we did together that has already given us some interesting results that we hope to continue to do as a way to feature what we can do together that we wouldn't be able to do without working together. Researchers in one sort of group, and then the patient advocacy and patient groups separate. I think we're already pointing to areas of research that we were not on our radar as a field. So Sheila and Sarah are here with me, and Courtney, whose face we're not going to see on the screen. Hi.
Hello.
Courtney, you look amazing. She has COVID. This is why
Thank
she's
you.
not here.
Yes, I'm local, but
We
unfortunately
can't
I
hear
have COVID.
you.
Oh, okay. Um, hmm,
Sarah, can you
Can you-
speak for Courtney? They're siblings. Telepathy, mind reading. Let's continue to work on that. I'll give you guys the mic and then we can continue to work on that. I don't think any other conference, anyone who would have had COVID would have done this. So this is just another example of how engaged and willing to participate people are. Sheila.
Okay. Hi, everyone. I'm Sheila Connor. My husband, Tom, was diagnosed in 2016 with Cadasil. 2015, he had suffered a pretty significant stroke one morning. We got him into the ER and they did a CAT scan, MRI, and they said to us, "When were your first two strokes?" And Tom said, and I said, "Well, he's never had a stroke before. He's really healthy." You know, he had none of the indicators of normal stroke indicators. He was 55 years old and healthy, went to, work out every day and always ate healthy, and just probably be the last person. When I called my family to tell them, they didn't believe me. They said, "Well, if any of us have had a stroke, we'd be like, oh, okay, but not Tom." So they sent him, they thought maybe it was AFib. So they sent him to a cardiologist, and the cardiologist called Tom his $1 million patient because they gave him every test possible and everything came up negative, and they just could not figure out what was going on. He had some white matter disease. There were obvious previous strokes. And then, he went through rehab. They said, "You're going to be here for 6 weeks." He said, "I'm out of here in 2. I cannot be in rehab." He was out in 2. We went on our way. A month later, he had another stroke. And this was in a different part of the brain. So he just kind of, they didn't know what to do except to give him occupational therapy, physical therapy, whatever he needed. But the cognition after the second stroke was pretty obvious. He would not be returning to work. And, at 55, young, active person, it was pretty tough. So about a year later, he was also having, he didn't say he was depressed. He said he was angry. He was angry about the situation. So we were referred to a Harvard physician, Harvard neurologist who was teaching at Duke. And they said, go talk to him and we'll give him some meds and he'll be fine. So he sat Tom down, listened to his story for 20 minutes and said, I'm sorry, but I believe this is what you have. And he wrote down Cadasil. He said, look it up or not, but I'm pretty convinced this is what you have and you need a blood test. So 3 weeks later, we got the blood test back. And that started our Cadasil journey. And Tom passed away in 2022. We have 4 children. They're in the consortium study. None of them have been tested, so they don't know their status. But I guess for us as a family, we are dedicating ourselves to raising awareness any way we can, being advocates for people in the community. It's so important. And so, we're joining many, many others in this community. I think of the Hunt family, the Kennedy family, the Carney family, Janet Orgarzo and her family, who have been doing this, trying to raise awareness on a local level for about, well over 10 years. And so I feel that we're joining some really good company to get it out there. And so what we're trying to figure out now is how do you get it to the next level? Yeah, local people know about it. How do we go national and really get this out there? It is, as we know, misdiagnosed. I mean, Tom went for a year. They just looked at him. They're like, we don't know, so how do we get it? How do we get it out there? The kids and I have started a business, Commonly Rare Designs, and we go to farmers markets, and my daughter Christine is a fabulous artist. So we sell her, handcrafted, one of a kind artwork. And we talk to people, can we talk to you about Cadasil? And we've gotten a couple of aha moments out, where people are like, oh, really? Can I take some information? So that's like kind of what we're trying to do. And, work with any group, Think Cadasil, CureCadasil, any way we can to get just awareness out, on a national level. So, people don't say, oh, really? What's that? They'll say, oh, yeah, I've heard of that. And that's the goal. So that's my story. So I'm going to hand it over to Sarah.
Can you guys hear me now? Hello.
Hi there, I'm Sarah. I live in Madison, Wisconsin with my husband. Where I work as a pediatric neuropsychologist. So I myself have Cadasil. My journey started in 2011. When my mom was suddenly developed pretty intense headaches and aura. Went to our local hospital, got a ton of workup. No one knew it was happening. Then was transferred like after a week to Northwestern in Chicago. They ended up finding Cadasil for her. And then we had more of an answer of why there had been so many generations of family members who had suddenly, for no clear reason, been developing early onset dementia. So then in 2015, when I was 25, I had been kind of noting for a couple of years that I had been having some word finding challenges. And then also thinking back around the age 11 or so, I had started to have headaches with visual aura. Didn't really think much about it. So then, knowing, kind of putting some things together, I don't know, I just had a bad feeling. So I decided to get tested, went to a neurologist. The neurologist said, hey, because you're so young and because there's no cure, I need you to persuade me why I should do this testing on you. And so I did persuade him. And then of course the results came back positive. So At that point, I started learning more about it. I've participated in several studies, the consortium, Dr. Elahi's study, and there was another one several years ago that I can't seem to figure out what it was. But and then I okay, thank you. There we go. So then I joined the community advisory group and spent some time with that, and this brings me here today.
You're on.
Now?
Yes.
Okay, hang on, because I'm getting some echoes. Okay, can I just get a thumbs up from the audience if you can still hear me? Okay, perfect. Thanks, everyone. Okay, so hi, my name is Courtney, and as Dr. Lahi mentioned earlier, I am Sarah's sister. I'm currently 31 years old. I live in the area, but unfortunately I'm sick, so I couldn't attend in person today. Just a little bit about my own background. I have a bachelor's degree in language and global studies with an MBA focused in data analytics. And I work as a data analyst in the travel industry currently. I try to stay really active. I love going on long walks with my dog and my husband, ice skating, traveling, hiking, reading. And I'm currently the newest member of the CureCadasil Community Advisory Group. I just joined about a month ago, so still kind of getting the hang of everything here. I was diagnosed with Cadasil in the fall of 2021. And unlike my sister Sarah, I really didn't have many or any symptoms. I chose to get tested just more to help with future family planning and just have a better picture of my health for now and in the future. Luckily, even though I didn't have any symptoms, I had no issues with the doctor allowing me to get tested. Everything was fully covered by insurance, so that was really great. Obviously, I found out that I was positive for Cadasil, which was a bit of a surprise. Obviously, I knew that there was a chance that it would happen, but again, I really hadn't experienced any or many symptoms. And I still don't think that I do experience many. Again, that was only a few years ago, and it's kind of hard to know for sure since I am quite young still, and some of the symptoms can kind of be easily written off. But in the past couple of years, I've gotten involved in multiple studies. So last April, I participated in the NIH Cadasil 2 study. And then I also joined the Cadasil Consortium study, I believe about a year ago. And then I'm participating remotely in Dr. Elahi's study as well. So, doing everything that I can to help raise awareness, help with research. And one of my hopes, just as someone, with a younger diagnosis, is to be able to continue to raise awareness for anyone, but particularly for people of a younger age. Obviously, for the majority of people, symptoms and diagnosis don't come until a little bit later in life. But, I think it's important to try to get those diagnoses for people younger so that we can just make decisions with the most information possible and live our lives as healthily and proactively as possible. Sorry, I kind of cut out on the other two intros, so I'm not sure how far everyone went. I'll stop with that. I know we've got a lot of other questions that'll kind of help us expand on other topics. So yeah, that's it for me for now. Thank you.
Thanks so much, Courtney. Can I have the slides back, please? Okay. So this is what we're doing. We think that the discovery of therapeutics has been fundamentally broken. Alzheimer's is the best example of that. But now, over the past few years, it's not because we're smarter than past researchers. We realize that, but it's because we have tools that they didn't have. So I think discovery of therapeutics is drastically flipped on its head, as opposed to going one hypothesis at a time and trying to piece together information that we don't have in humans to formulate that hypothesis. We can actually start formulating hypotheses based on the large data and then use all the toolkits that we have at our disposal to test whether our hypothesis is correct or not. And that discovery cannot happen in animal models. It ought to happen in human beings. It also cannot happen in brain tissue from autopsy cases. That's an end stage of the disease. It ought to happen in people like us, across the board of age, across the board of disease stage. And so it's fundamentally important to participate in research to donate biospecimen. That's the most important component in our research. We are actually pretty minimalistic with respect to what clinical data we're gathering. The NIH study is doing a much deeper dive into head to toe clinical measurements. The consortium also has more measurements. And so those studies will determine whether we have the right measurements for our clinical readout. Based on an initial study done 5 years ago, we think executive function, processing speed, fine motor function may be good enough. So that's what we are focusing on. What we do is a really deep dive in blood that we are getting. We're not collecting cerebrospinal fluid because we don't think it's scalable, although at some point we may need to. We're not going there yet. And I'm saying paper, that's not our goal, but it means concrete results, obtained. Every paper that we've published has been done in blood. We are seeing in blood a signature of disease, and we see that signature change across the disease spectrum, which is why it's really important to start looking early. Because ultimately, the most impactful therapeutics would be before people are symptomatic, although we think that the target of therapeutics may change as disease progresses, and we hope to at one point have therapeutics available to whoever, no matter what stage of disease. Why? Because a lot of people get misdiagnosed. Not all diagnoses are going to be early. This is still a problem of awareness, but it's also a problem of phenotype, meaning that individuals that carry Notch3 mutations don't all look the same. And so you have some patients who get misdiagnosed as frontotemporal degeneration. Some other patients actually have Parkinson's disease. I don't know if it's a misdiagnosis, but they have classical looking Parkinson's disease. So the biology, it's like a before and after moment. The biology of Notch3 is very complex, potentially in a good way. It means a lot of various therapeutic entry points into perturbed pathways. But that's also what you would expect for blood vessel disease. This is not a disease that's affecting neurons, first and foremost. That is downstream. So not only it gives us a time span during which we can intervene, but it also means that whatever that individual is going to look like as the brain degenerates is not necessarily driven by the Notch3 mutation, which is why there's such diversity. So together, the NIH study, the consortium study, and us have the task, and our European colleagues, I think, have the task of coming up with staging biomarkers such that we can determine with greater precision than flip of a coin what stage of disease from a biological, pathological perspective an individual is, and don't just depend on the clinical phenotype. I think that would be really important. So that's why the almost transparent human, I need to change the color, that human, because the discovery needs to be made in human beings. What we do after that in our study is we have derived stem cells from patient-derived lines. We've corrected the mutation. And then this is, I think, the revolutionary tool that we didn't have 10, 20 years ago. We can derive cells in a dish. We can also derive more complicated models, vasculature. We can derive vasculature connected to neurons and astrocytes. So we can ultimately create mini brains. In fact, some people call them mini brains. They're like little balls that represent all the different cell types that are in the brain, and understand whether what we saw is correlating in the blood of humans actually gives us defects in a dish. And that's a starting point of screening drugs. So the biology has a blueprint, it has a pathway. What doesn't have a pathway is what do we need to understand about the experience of the patients. So as I was getting incredibly excited about the approach that we can take to discovery, I realized, but what are we going to treat exactly? What are the symptoms? What does the journey look like from the start to the finish? And that's when we partnered with the patient advocacy groups and the patients to tell us. And Sheila and I spoke weekly at some point. We communicated a lot, and we had the advisory meetings with CureCadasil, and we thought, let's ask everyone. Let's create a survey. All the work was done by Sheila, Andrea, Key, and others on the community advisory board, and ask what bothers people most. This is just showing the cohort. This is actually the slide I used last year, and we have not updated it. So it's more people. More people are actually participating in this research, and that is absolutely amazing. One, the disease is not as rare as we thought. That I think we all know. And two, people are willing to participate. We're not paying anyone. I think we should actually compensate for time, but we can do this. We can do this together. It's absolutely incredible how engaged the community is. So what should be the outcomes? This was the survey, and there's a Project Cadasil that distributed the survey that was drafted by Sheila and Andrea. Key Cole really spearheaded the circulation of the survey. And so Facebook, I think, is where most people are, less so the other platforms, but and this is the regionality, very, very US heavy, but this was English. So we did bias it to English speaking individuals. Oh, and I should add that this was open for 1 month. It's open actually, and there's a QR code at the end of it. But this was results pulled over just 1 month, and it was about 200 people that responded. And I think we have more results that we can potentially analyze and draft. And this was just the US based participation. Age and gender distribution, heavy on the midlife. And I think this is also a bias of computer access and such, and maybe more time or also symptomatic presentation. More women, not shocking. This is always the case with all observational research. Now, what was the most bothersome symptom to individuals? What do you think? Memory? Migraines. Jane is cheating. Jane. Okay, yes. Fatigue. I'm sorry, that's a little bit small. So on a scale of 10 to 1, and some NA, some people did not respond, we asked individuals to rate what the most bothersome symptom was for them. And by far, the most bothersome symptom was fatigue. I didn't know this. I had zero idea of this. And then strokes, not surprising. And then headaches. And then mood problems. And maybe mood problems is here because we expect it midlife, don't we? And for the state of the world, we do. But mood problems are actually very common. Apathy, interestingly enough, is last, but I think most people suffering from apathy don't know necessarily. So this is an ascertainment bias, potentially. this is really important. Pain is underappreciated, but a lot of my patients, as disease progresses, do have pain. And this, we need to have biology. I just met with a company that is interested in collaborating, and they're interested in so many of these, but they think of them as separate. And the selling point for Cadasil and why people need to go after Cadasil, drug developers, companies, industry, anyone who is interested in developing therapeutics, is you have so many of the symptoms that most diseases have one or two. So if we find, and it's monogenic, so if we find therapeutics, we are going to help individuals with Cadasil and others suffering from sporadic disease. This is just the demographics, and this is also very interesting and speaks to the undiagnosed. Most of the symptoms that bring someone to the medical attention are migraine and stroke and things of that sort, for which someone gets an MRI and then gets this sort of puzzled why so much white matter and now genetic testing. So I'm not sure about skin biopsy because I don't think we ever do it anymore. Even in the setting of variant of unknown significance, which is very unsatisfying for individuals who get that. I think when you have a high a priori, if you have the white matter changes, if you have some symptoms, then the variant of unknown significance no longer has unknown significance. That is why these symptoms. So we are also trying to change that. So the other component of this, and this is unfinished business, but I think we have been talking a lot, and I am growing increasingly unsatisfied of observing our cohort. I'm incredibly appreciative that people are coming and donating blood for us to continue the biological discovery process. But why not turn our observational study into a clinical trial study, not with new drugs for which we have a lengthier pathway to tox and making sure that we're not going to do harm. But I realize that individuals who see me in clinic are testing things on themselves. Hearsay, this and that. And most of it is quite benign. So, and what I do as a physician, I say, I don't know, actually. And it's quite embarrassing that I don't know. I mean, I can't tell people to go off aspirin or stay on aspirin. Someone just sent me an article about aspirin not being very helpful for people. And we know the effect sizes in cardiovascular studies was very small and large sample sizes and such. But I don't know, maybe it's actually helping Cadasil. I don't know. And so there are a lot of questions to be answered. And the starting point here was we wanted to see what people are testing on themselves. This is not comprehensive, but we are going to continue to accumulate this data. It's not a statistical analysis necessarily that we're going to do. It's very qualitative, but we want to know what people are testing. So here, most of the prescriptions by physicians is for vascular things, which is not very surprising. You do want to optimize blood vessel health. But the question is, how do you optimize it in Cadasil? And that's not evident at all. So this is one. And then a lot of vitamins, L-arginine may be something that individuals without Cadasil don't necessarily take. The rest of it, I think, are all things that most people try or take for other things. CBD may be coming from California. I don't know. I think every single patient was taking some sort of variant of that. And then other things. And I think we can, at some point, if the NIH study is interested in this, if the consortium is interested in this, the next iteration of all these studies could be clinical trials, because you still get very good data. In fact, potentially better data because there's more tighter follow-up. And we may be able to answer some questions, some things that we could potentially test. And then non-prescription over-the-counter aspirin. But I want to add here that many of my patients are multiple agents because they were placed on them before anyone knew they had CADASIL. And they had a lacunar stroke at age 30, and someone said, oh my God, your blood vessels are in awful state. We need to go after this hardcore. But they just have a Notch 3 mutation. So should they stay on the dual antiplatelet? Is that risky to be on multiple things? Clopidogrel or Plavix and aspirin? I had one patient on an anticoagulant. And what do you do as a physician? So I see CADASIL patients all the time. That's all I see. But imagine a neurologist in the community who doesn't. What can they do? It's very uncomfortable to take someone off a medication, and for them, uncomfortable is a complete. It's a very small word for how you would feel if they have a stroke after that. And despite the fact that I'm a CADASIL expert and I do science that's CADASIL driven, I don't know what to do. And I hesitate and I communicate extensively with that patient before I change their medication because I don't know if it's helping them. Maybe it actually is helping them. So all of these are going back to the conversation that we had. At the very least, we could pull data from medical records across health systems that have cared for individuals with CADASIL and look at this at a larger scale. We would have better numbers than the N of 10 or N of 20 or 30 that each practitioner would have to see what are the trajectories that people have when they are on these medications and when they're not. Then, we are talking about pharmaceuticals, and I think the world of Alzheimer's disease is actually moving towards more interesting therapeutics, not just drugs, but some stimulations and such. And I will show in a few slides how stress is one of the worst triggers. And so what do we do for stress? Aspirin? I don't think so. So what else do we need to look at that is non-pharmaceutical for the community of individuals at risk for CADASIL outcomes? So meditation helps with mood and sleep, and interestingly enough, cognitive fog, which I found that to be very interesting. Here, I want to take a brief pause and ask our panelists what they think. Have their loved ones or themselves ever done this? And how should we go about stepping into a more holistic approach for increasing the impact of any kind of therapeutic that we may have for this community?
Well, I'll just speak to Tom when he had his first stroke. 2 years leading up to it, he had a lot of upheaval in work. He was transferred down in North Carolina, which was lovely, by the way. But there was a lot of stress. And I think stress will kill some people. It can kill people. So he was doing, working out. He loved music. He was a musician. He loved, just being with the kids. That's kind of how going to Walt Disney World was his way of decompressing. But, the scale kind of tipped. And when we were at the neurologist who diagnosed him, Tom said to him, I'm very concerned. Tom didn't want to talk about himself. I'm very concerned about Sheila and how she's going to handle this. So the doctor just looked at me and said, what do you do for stress management? I said, well, I'm a newly Reiki master, so I practice Reiki twice a day, and I also practice meditation twice a day. And he said, okay, Sheila, you're going to be fine. Let's talk about you, Tom. Trying to get Tom, to medicate. He was always on the go. So getting him to sit still was pretty hard. But I think definitely it helps. Whatever your thing is, to get out there and whatever will relieve the stress. When he was relaxed, he seemed to do better. If he was anxious or upset about something, okay, how can we bring that down? And I encourage my kids, they're ages 33 to 21, you know, this is a lot. It's kind of a daily part of our life. They're very involved in the awareness end of it. But everybody needs a break. So if you need to step back, but make sure you have something that's just for yourself or a little group, or they're very supportive of each other, but everybody needs that break. So if you can find something that, you know, you can do to kind of just bring your shoulders down from up here to down here and relax, I think we have found very helpful.
Yeah, I think it's interesting in the, at least I can speak for maybe more Western society, we are really focused on medications and such. And of course there's a place for that. But the stress relief, I mean, it makes sense. I don't know, our Western society, we're very focused on like busy, you know, you got no rest. Rest is for the weak or whatnot. And I mean, it's backwards. And I think this is just very important to take some time to, you know, incorporate these relaxation techniques into your, you know, everyday life practices. But I feel like in terms of thinking about meditation, that's not going to be very natural for any of us. Like I've tried to do it myself, and then I'm like, oh, but I have this other thing to do, and it's super hard. It takes a lot of practice. And so, you know, I think if there was more focus on meditation practices, and it would be helpful to have like classes and like actually like going out of your way, like here are resources. You're not just like do it, bye, but like here is how I can provide some resources for you to help you learn how to take these how to relieve stress.
Everyone here?
Meditation stresses me out. Go ahead, Courtney.
Okay. So, you know, in addition to obviously, you know, trying personally, I haven't had much success, excuse me, with meditation. I find it very easy to get distracted, but I do have other ways of coping, just working out or maybe yoga or something like that. But I will speak to the possibility of potentially seeing stress affecting just my mother's symptoms. We've had a really stressful, you know, year or a few months in our family due to all kinds of various things. And well, a couple years, I would say, for her. And again, it's hard to know for sure, but she definitely does not have good stress management skills, good coping skills, anything like that. And she's also definitely experienced some just cognitive decline, losing some of her executive functioning. And so, you know, again, who's to say if it is correlated or if it's just coincidence? We can never know for sure, but it's certainly interesting to see when there is that lack of ability to deal with stress and just kind of calm oneself down, how there potentially has been an increase of symptoms.
Thanks so much. That last point is really critical. A different paper, different project, not in CureCadasil, that we did show that perceived stress is what has biological underpinnings. If you look at inflammatory measures or immune dysfunction measures, it's the perception of stress. So you could be on Wall Street in that lovely, cozy environment, but your mind is on the beach somewhere and you're doing things, you know, one thing at a time, but you're not necessarily perceiving stress. In mice, it's the feeling of defeat. So it's when mice are defeated, the social defeat model. So you can control this. So it's not about, is my life perfect, or I have 5 kids. That's a little stressful. It's how you perceive it. So I am no one to give advice about meditation or stress relief, but I do think that there is a way for everyone. But here we just wanted to highlight that one factor that is modifiable is not something that we will go after therapeutically, but incredibly important biologically. And so each person's stress relief is going to be different. because it depends on who you are. But maybe this year, this is something that you can walk away with and invest in your own life. Someone may do kickboxing, someone else may go for a walk, and someone does hangs out with friends. We each have our own sort of exit from that state. This could be a trial too, by the way. Our trials don't need to be pharmaceutical, but it's a harder thing to trial. And triggers. So this really, again, emphasizes that. And many of my patients who stop working is because of the feeling of stress. Sleep deprivation, I think, triggers any human, no matter what. But the two are very related. Stress may have such a great impact because it's not something that you leave when you sleep. It's a state. It actually impairs sleep. It's impacting so many different aspects. It impacts the daytime and the sleep moments. And here I also want to take a pause and just hear a little bit about sleep from the panelist. How was sleep for your husband? Yours, I'm assuming, was amazing with your meditation practice, but tell us about sleep.
Do you want to go first?
Sure. I mean, very briefly, and my husband can attest to this. I do not do well when I have sleep, don't get good sleep. Now, me being younger, not having as many symptoms, I personally haven't had like an episode per se, but I definitely, like most people would, have much more brain fog and challenges expressing myself, and probably slower, processing that day. But certainly I can tell you that poor sleep affects me more than him. So I don't know.
Yeah, and I think for Tom, he was always, I guess, a good sleeper until the first stroke, and then he could not sleep at all, and he couldn't shut his brain off. He said, I can't shut it off. My brain is just going all the time. I can't quiet it. So he did try the meditation, mixed results. They did put him on medication. Unfortunately, he was allergic, and then he was getting some really good sleep, but then he became allergic to it. So it's like, well, so then it was a mixture of medication. So what's more important? They took him off everything and then started adding things back in. So it was tough, but finally we did find a medication, and that would just help him sleep.
And then the other aspect of this is to just get your sleep checked. So if, the question is always, do you snore? Do you this? Do you that? But it's a modifiable component of your life. And my sleep colleagues say 1/3 of your life is spent sleeping. So if we can modify the quality of that in any way, then it could also impact the brain health.
Yeah, I would just add, I guess, I've always been someone who's more affected than others. I felt by, lack of a good night's sleep. And I also do experience restless leg syndrome, potentially correlated to stress. It's hard to say for sure. It kind of comes and goes in bouts at a time, which obviously is a huge issue with trying to sleep. And I have not had this checked out, but I do feel as though I might have a bit of sleep apnea. It's not anything that, as far as I know, is affecting me much at this point. But again, still young enough that I'm relatively asymptomatic, haven't had a stroke or anything like that. So it's very, early on, kind of in the journey for me. I don't know how much of that is affected by stress or not, but it certainly causes stress. And then in turn can affect my sleep after that.
Courtney, you need a sleep study.
Yeah, I know.
Can we have the slides back, please? And then this. So what should we go for? And obviously cure is what we want to go for. But while we're working toward a cure, we need some treatments. And there was a response for treatment in general, but slow down, prevention, and improvement of symptoms. And improvement of symptoms, I always thought impossible. I mean, you can't, like, once you have damage, but it's not true, actually, because some of the symptoms of CADASIL are such that it sort of snowballs, but it's not necessarily an irreversible avalanche. And so, as such, I think we can shoot also for improvement. And this is a phase, so we want to hear from you. This is a phase where we are really, really actively thinking about trials. And so let me go to the next slides. Participate in the survey and or email. I'm just going to leave this up for 2 seconds. Well, a little bit more than 2 seconds, but there is also an email address that we want you to email your suggestions and questions to us. And if you think that the survey is not capturing some important aspects, also suggest that. This is to thank everyone. This is a snippet of individuals working and contributing to our project. Ruth Axon and Harrison Chan are the clinical research coordinators at our site. Mariana Lemos Duarte is a senior scientist who pulled the data and is going to pull the data again and create newer figures. You guys, the research participants, both patients and families, it's been incredible to work with you. The CAG, it's been a true honor to work alongside you. And thanks to CureCadasil for pulling this community together and channeling our efforts. Thanks to Chain Zuckerberg for the dollars. And this is to participate in our research. And that's an email address that we created so that it's not human or person dependent. It's going to be permanent, even when the research coordinators go to graduate school, which happens a lot, thankfully. We will, that will be a permanent email address that you don't need to change. And I'm going to go back. I don't want to reveal the next talk. Thanks so much for your attention.
I'm sorry. Oh, okay. All right. And just finishing up, I just wanted to say to the CADASIL community out there, Tom had no other family. He was adopted. So we were learning from scratch. And I don't know if this is a good thing or a bad thing, but in the course of a year, I took him to 5 different neurologists because I didn't like the answers I was getting. And I finally hit upon someone in Rhode Island who sat down with us and the nuts and the bolts of what was happening, what could happen. And it took the fear out of it when you don't know about this disease and you see someone who is so healthy and strong and go, and then all of a sudden they're not. It's incredibly frightening. So I guess I would just say to the CADASIL community, you know, use your resources. Some people have to advocate for themselves, but use us, then use the community, reach out to us, either, you know, through social media. You know, he had 2 primary care physicians and they both said, they had research in their background. So they said, we know nothing about this disease, so we're going to learn with you. And that's fantastic. You know, we were so lucky. So, you know, find doctors you can work with. If you can get involved, please do get involved. Lots of things you can do. And, you know, go to CureCadasil or Think Cadasil and let's spread the word. So thank you. Thank you, Dr.
Thanks so much. Harrison, do you mind standing up? He's actually in the room. So Ruth is not here, but Harrison is there. Harrison is going to medical school. He does bench work, but I wanted to take the sort of clinical work that he's doing with CADASIL away because he had a lot of work and he said that's the most rewarding part of his work. So he really, really enjoys all the interactions that he's had with you. And contact him if you want to participate.
So great. And there'll be time later at the end of the session when we have our panel for discussion for more questions. I'm sure that people have. It was a really, really nice session. So if you have an old program that we circulated for some time and posted, this was going to be Jose Biller, who was speaking on behalf of the Coruscant, or the study consortium study, but he was unable to. He had a conflict that came up at the last minute, and so we're equally fortunate, though, to have Jamie Elliott, who is also one of the investigators, site principal investigators up in Madison for the consortium study, which is really exciting. We'll maybe not get a lot of results in, but really when you see the numbers going up, it's really heartening. So, Jamie.
Thank you all so much for having me. I'm a vascular neurologist at the University of Wisconsin-Madison, and I did my early research on cognitive decline, commonalities between neurodegenerative disease and sleep. And so I was always interested in cognition and sleep. And then I loved stroke when I did my clinical work. And honestly, many people didn't think they go well together, but I think they go very well together. And especially it's a good fit for people with these particular problems. But also the overall population has small vessel disease as well that also contributes to stroke and cognitive issues amongst everyone. So that's how I came here. And I'm just giving you a very brief little update on the CureCadasil consortium. For people, I just wanted to say about the earlier interest in meditation. I think that's really important. The University of Wisconsin has a Center for Healthy Minds that does a lot of research into kind of alternative treatments for stress management. And for those of you who are thinking they wish they had a guided form of meditation, they have a great app that is data-based if you wanted to download that. So we'll just briefly kind of go through the study, and the update is just about enrollment. We're not quite at the data time yet, but it's a 5-year multi-site observational study tracking people with Notch3 pathogenic variants and controls from families with Notch3 pathogenic variants. And our goal is to enroll 500 people. These people will have 3 in-person visits with a number of different assessments. So those include kind of an extensive interview about their own past medical history, the medications that they're taking and have tried in the past, various assays of their mood and migraine and other related symptoms, a neurological exam, some cognitive testing, and then a blood draw and MRI to look for a number of different biomarkers. What we are hoping to do, as Dr. Elahe covered very extensively, and the idea of this kind of research is multifaceted. So on the one hand, as you touched on, people with these pathogenic variants have a lot of diversity in terms of their symptoms. And it'd be nice if for individuals, we had some way to guide them about what to expect in the future. Just as we mentioned, like where are they in the time course of their disease? What might they expect? So having a larger group of people with a lot of information about the way that their disease presents is one way that we can do that. Trying to even define who's symptomatic and not symptomatic is not straightforward at this point, depending on what type of symptoms you decide. And so seeing if people's personal experience of their symptoms correlates with any of the things that we can measure in a way that can meaningfully then be used to find interventions. Just as Dr. Elahe was interested, I am also very interested in the fact that we have a big diversity of adaptation of normal vascular risk management. And the same way we'll be able to see if there's any kind of readout in terms of the medications people have been exposed to in terms of benefit, and also an idea for testing known drugs in the future as we work on new drug development. And then essentially looking for readouts that we can use in a small source of time for these kind of treatment trials, the things people care about. Other than things like fatigue, a lot of them are either rare events like stroke that you have to wait a long time, hopefully, for people to have, or things that elapse over a very prolonged period of time in terms of decline of cognitive function. So those are too long for most types of treatment trials. So we need outcome measures that are shorter so that we can start to see if things are working without having people on a medication for decades to see a benefit. Oh, the final thing I didn't mention is we do like to get some an interview with a companion, which is either a family member, partner, friend, which is common practice in studies that look at things like cognitive performance and mood, because people may not, as we notice them themselves, things like apathy, and also comments on how functional they are at home. So we have a number of sites across the US that you can see on this map here, and I am one of the site PIs in Madison, which is fairly close to here. In terms of people and joining the trial, there are some things that are beneficial for people that decide to join. So anybody can join if they know their Notch3 status. But we also have people join that don't know and provide the genetic testing. So anyone who's from a family with Cadasil can join and be tested. People that don't know their own genetic status will talk with a genetic counselor about that. It could be a very fraught decision to decide if you want to know or don't want to know. So they can kind of go through the pros and cons of that. It's a nice way to get this testing done because, of course, it is all only within the study that the outcome would be known. So it's not released to your insurance or your other medical providers if those are things that concern you. And even within the study, if you chose not to find your results, like none of the people you interact with will know your results. So there's no chance that you will accidentally find them. In terms of enrollment, we have enrolled over 300 people, which I think is fantastic for a rare disease. And as you can see on this chart, the kind of slope of enrollment has really increased. I think everybody that was involved in clinical trials at the tail end of COVID knows it was a little slow for a bit there. But we're really kind of ramping up. So we're looking for about 200 more people. So the focus is really on that for the moment, which is why we don't have a lot of the data outcomes, but hopefully by next year, we'll have some more things to show you there. People are eligible, like I mentioned before, if they're adults, if they themselves have a pathogenic Notch3 variant or a diagnosis of Cadasil, or if they have family member that has it, and either they know their status is negative or they don't know. So all of those people are potential participants for us. And then ideally, of course, you'd be willing to do all the various assays, although there are some exceptions made if some of them don't sound appealing. We just a little comment on people that are non-carriers for the pathogenic Notch3 variants. Of course, doing any research is incredibly selfless. You don't get a lot out of this type of observational research, and it's been amazing how many people have joined and how willing they are to do that. But it's a little more direct when you yourself have the abnormal gene variants. But we need the controls because just like we talked about a lot here, it's very difficult to tell if some of the symptoms are just like being a normal human, like you're fatigued, you have memory issues, you have mood problems. And although it's frustrating that the disease is misdiagnosed, all the symptoms are things that other people have, right? So to make sure that what we're finding is truly related to the gene variants, we need to have controls. And these family members that don't carry it are a great option because they otherwise are genetically similar, may have similar environment that they grew up in and kind of similar backgrounds. So if for anybody that may be listening that are one of the people who joined in this situation, we really appreciate it. And I think a lot of times people don't realize that they can also be an important contributor to research. So just because, of course, it's burdensome to do any research. Even if you live right next door to our hospital, you don't want to go to the hospital on your day off and do a bunch of studies, especially an MRI. But we are happy to kind of coordinate travel for anyone that needs to come to one of our sites, whatever's most convenient. And a really nice thing that we've been able to do is for families that live in different areas is to kind of coordinate them all to come together for visits. So it's like a little family reunion. So you get a little benefit there as well. We are excited, of course, about the baseline set of assays that we're running, but also excited that we've been able to add more things to this kind of big group of Cadasil patients, and we're very happy to be working with other studies. There's just a few examples. We have MRI technique that allows us to look more detailed manner at the vasculature of the brain. That's been approved and funded, so we're doing this now as a possible MRI-based biomarker. We have another MRI test that looks at the responsiveness of blood vessels in response to CO2 challenge. And again, of course, it's a blood vessel disease, so we're just trying to see what we can pick up in terms of early abnormalities. We also submitted a grant looking in the eye. So neurologists will always tell you the back of the eye is the front of the brain, but all the blood vessels and neurons there can allow us to look directly at those structures instead of kind of these indirect routes of imaging for the brain. And then I'm particularly interested in adding on some wearables. I've always been interested in sleep, and this is a nice way to look at people's sleep quality in their own homes all the time instead of just an isolated test in a sleep clinic. So more things coming in the future, and participants in our study can decide to do these ancillary studies as they're interested. We are so happy to be part of this community, which has been great to meet as I meet people that are joining the study. And we do a number of webinars, which are all available on the website. We've just started this, which has been nice, which is a call with one of our site neurologists that's just available to people that are in the study. I think it's interesting, of course, to find someone that has heard of the disease, right? And that could be, we always let people that come to see us at our sites kind of talk to the neurologist to see if they have questions. And this is just an opportunity to bring yourself and anybody who's interested to meet with a neurologist that's familiar with CureCadasil to ask any questions you may have about it generally and the study. And we're doing those mostly via WebEx, so you don't have to travel for that. And then, of course, you have the community engagement session this afternoon, so please don't miss that. We'll be there. And that's all. So I'm happy to take any questions about it at the roundtable. Thank you.
That's great. Thanks very much. Okay. So we're fortunate to have an update also of the NIH NHLBI study. And Lisa came all the way, and believe me, I know how much paperwork and red tape it takes for her to escape the NIH and come out into the field and actually talk to real people. So she's well established as an investigator in NHLBI, and they've been pursuing this study first as a pilot and now as a more of a full-blown study, which she's going to give us an update on.
Yeah, so first thing, I actually wanted to echo what Dr. Elahi had said. I think this is an amazing conference, and I'm very thankful to be here. Also, my direct supervisor, Dr. Baum, came last year, and that triggered a number of collaborations, including work with Dr. Elahi. We're continually talking and figuring out how can we do things together. Yeah, so I joined NHLBI, National Heart, Lung, and Blood Institute, back in 2016. And at the time, I was a clinical program manager. And that is exactly when the CureCadasil 1 study had just started, the pilot study. And I have been there 8 years, and I have since become the principal investigator of our second CureCadasil study, which is an expansion of that effort. The team was really small at the time, and it has grown exponentially as we've learned more. To give you a little bit of background, National Institutes of Health is a federal government agency. So we are funded by Congress, and we have the clinical center, which is this picture here, that includes a number of different institutes focused on different things: the eye, cancer, the vasculature, which is what I'm a part of. Our patients come from all over the US as well as internationally. All of the travel and lodging is paid for. Every patient that comes is enrolled in a protocol. We are a research hospital, the largest in the world, and we don't have an emergency department or a maternity ward. We're exclusively focused on research. And one of my favorite things about the clinical center is that the labs are surrounding the clinics, so that the information we get from the patients informs the research we do in the lab, and the research we do in the lab informs how we care for our patients. Really, our primary mission is to use the science that we perform in the labs to inform and advance human health. And that is one of the reasons we've become the lead in the world, really, of translational medicine and going from the bench to the bedside. So for CureCadasil, back in 2016, we assembled a fairly large team. Oop, wrong button. A fairly large team. NHLBI is up here. But we also have investigators that are interested in the neurological findings, psychometric. We do a lot of imaging for the brain. We're very interested in the functional genetics and how the particular variants inform what happens to that protein. And we do a lot of vascular diagnostic testing. All of it is non-invasive, so they're tests that are really just imaging. But as Dr. Elahi said, not just focused on the brain, we're looking at the entire body. And we do have an investigator here in Northwestern in Chicago who is helping us with the basic research, so a lot of the mouse studies and things like that. And so you guys are people that know all about CureCadasil, so I'm not going to spend a ton on this. We know that it's due to NOTCH3 variants. We don't know what the molecular mechanism is. In the past, it used to be diagnosed by electron microscopy images of skin biopsies. This is actually one of our CureCadasil patients from CureCadasil 1. You look for blood vessels like this, which you can't always find in that skin biopsy. And you look for these little deposits called granular osmophilic material, or GOMS. I was very excited when I got this picture on the microscope. But that was not a great way to diagnose CureCadasil, because we were missing people, and some 25% of people with NOTCH3 variants don't actually have GOMS. We do know that it's a small vessel disease, but we think because NOTCH3 is expressed by all smooth muscle cells and parasites, this is a parasite, throughout the body, there are definitely other areas that are potentially affected. We talked a lot about the different symptoms and when they start, but really the hallmark is the white matter hyperintensities that you see on brain scans for patients with CureCadasil. And we don't have available treatment options that are specific for CureCadasil. We really just do a lot of management for lifestyle changes and controlling risk factors. And so an overview of our two studies. Our first study was a pilot study. Like I said, our team was really small at the time. We brought in 20 patients yearly for 3 years, a short period of time. Those patients were within 5 years of CureCadasil diagnosis, so pretty early on in their disease presentation. And I have to say, because we're looking for people that have a genetic diagnosis, we're really getting people that are having clinical symptoms of CureCadasil. And we know now that more people than we know out there have NOTCH3 variants that are maybe completely asymptomatic. So that part of the population doesn't have a reason to get tested for NOTCH3 variants. And back then, we were really focused on better understanding CureCadasil and what's going on there. That study completed and is currently in analysis. I'll show you a little bit of that. It is data that we're hoping to publish very soon. So it's a big set of data. So I'm only going to give you a bird's eye view. But we've expanded to 100 patients with NOTCH3 variants or CureCadasil. And I say NOTCH3 variants because some people may not. There is some discussion that some of these NOTCH3 variants don't actually lead to CureCadasil. We are also recruiting 40 healthy volunteers, and this was something that we were kind of doing beforehand as well with pilot study. But like Jamie said, we recruit spouses and companions that travel with our CureCadasil patients to serve as healthy volunteers, and we've continued to do so. And the advantage of that is that we're actually getting a group of people that tend to be age matched to our patients. So I don't know, for those that have been involved in clinical studies, we compensate for the experiments and the testing that we do. And oftentimes when we put those studies out there and ask for healthy volunteers, we get a bunch of 20-year-olds, which is not helpful. But yeah, so we do pretty comprehensive evaluations, like some of the other studies, your typical HMP, as well as get clinical labs, research labs. We do biospecimen collection. We focus on the skin biopsy, although we can do lumbar punctures. We have a genetics, a neurology, and an ophthalmology consults. We're very interested in those eye findings. And we do a brain MRI with a fairly extensive set of cognitive testing, as well as a number of non-invasive vascular studies. And so switching focus to the CureCadasil 1 study, I wanted to give you guys a little bit of an overview. And I know some people saw this at the webinar as well, but there's a couple of things I want to point out. So you'll notice here that instead of 20 patients, we ended up with 19 patients. That's because one person presented with a NOSH3 variants. But when we looked at their brain scan and their symptoms, they were very different from CureCadasil, and we weren't sure it was actually CureCadasil. Turned out that person had West Nile virus, as well as an asymptomatic NOSH3 variant that both their parents carried. So 19 patients it is. Pretty even distribution at the time of males versus females. Our population is largely white. All of our neuropsychometric testing at the time is in English. So we're not capturing, I think, some of the minorities in our cohort. And then when we talk about risk factors, what we see a lot is the high cholesterol, but sleep came up earlier, and I've actually been crunching the data, and sleep apnea should be listed here moving forward. About 33% of the people in the study had sleep apnea. And with CureCadasil 2, we're seeing that as well. So they have a confirmed diagnosis of sleep apnea. That sleep test is actually pretty important. We do know that a lot of people have sleep apnea, but it could be that this is one of the risk factors that we can control for in keeping a brain healthy for a person that has a NOSH3 variant. And then with the medications, you see here antidepressants and anxiolytics come up a lot, and mood controlling medications are also something that can help with the stress and some of those other things. So from that, I'll start back here then. When we look at psychiatric symptoms for CureCadasil 1 cohort, anxiety and depression were the two things that came up the most. A lot of our patients had strokes coming into the study. Some of them didn't and did not develop stroke at that time, again, kind of early in the disease. Some of the patients did develop a stroke between visits. They had mild cognitive impairments, and many people suffered from migraines with or without aura, oftentimes very complex migraine presentation. And then one of the main things we're doing for this particular cohort of patients is the brain MRI quantification. This is one of the reasons that so far we've needed for patients to come to the clinical center, because we need to control for the magnet that we use for brain scans, as well as for the sequences that we're using, so that we can quantify the white matter changes that are happening in patients with CureCadasil. So when you look at this mask here of the brain MRI, the red shows you the white matter changes. And so for this patient, for example, who had no strokes during the study visit, when you look at visit 1, that's their baseline scan, and compare it to visit 3, there is a small amount of progression of the white matter disease. When we measured this patient, it was about 13% increase. When we look at a patient with strokes, we're not only seeing those changes between visit 1 and visit 3, where you see an increase that was closer to 30%, but we're also detecting a flare up here where around the time that they were getting ready to come, they had a stroke, an acute stroke. And right after their visit, they had another subacute stroke. So you can see that at visit 2, their white matter changes really flare up before they settle back down. And then for neuropsychometric testing, so all the cognitive testing, we look very extensively at a number of domains. And we also obtain an average score for our CADASIL patients. And with this cohort that, as I said, was fairly early on from their diagnosis, we see mild impairments, but already we're seeing some changes on the psychomotor domain, which seem to be the most affected. So quick summary, what we found from that pilot study is that actually the disease progression was fairly slow in the study period, those 3 years. All of the patients, regardless of where their Notch3 variant was, and that was a pretty varied distribution of Notch3 variants, had the typical white matter changes of CADASIL. Like I said, they had very variable genetics, that they were within many of the different regions of the gene. And that seems to have been reflecting a fairly variable clinical symptom presentation. Some people had almost no symptoms and some people had significant symptoms. For those that had almost no symptoms, they had come to a CADASIL diagnosis either for family testing, like a parent or a grandparent, or for family planning, knowing that they had a parent with CADASIL. On the neurological neuropsychometric, we found that balance was a bit impaired. Patients with CADASIL, verbal, visual learning a little bit, and psychomotor domains. And a lot of the other data is currently in analysis. I'm really hopeful that I can get a manuscript out in the next couple of months that I can share with all of you, hopefully through CureCadasil. And so to shift to the CADASIL 2 protocol, we took all those lessons that we got from CADASIL 1 and applied them to CADASIL 2. So we realized that 3 years was not long enough to follow patients. So we went for a long-term 9-year study. The patients come every 3 years, and it's a total of 4 visits. We think of it as a long-term relationship between us and the patients. So I hope you like us. But we really want to be a home for patients with the Notch3 variants in CADASIL. And for that reason, we have a fairly large team of NIH experts that are not just focused on the vascular side, but also on the eye, neurology, imaging, and all those other things. Our testing is comprehensive, and it's tailored to CADASIL first and foremost. So we're using cutting edge technologies and everything that is available to us at NIH to be able to quantify CADASIL and its progression. But we also have the ability to tailor to individual patients. So for example, that person that came up that looked very different than an actual CADASIL patient, we were able to enroll them in another study and help them figure out that they had West Nile virus so that they can go on and get treatment and be seen for that particular reason. And then all of our clinical data is made available to the patients and their physicians, because really our goal is to serve as long-term consultants to help with the patient care. And of course, the ultimate goal, aside from understanding that disease mechanism that maybe can give us some molecular targets and help us find biomarkers, is to identify treatments for CADASIL. And at NIH, we're kind of the experts of taking medications that already are approved and repurposing them for different things. Our team doesn't only study CADASIL, we study other rare genetic conditions that have a vascular component to them, and we've done this a lot. But we want to have an interventional clinical trial at NIH, and we want to collaborate with everyone else that is doing the CADASIL research so that we can really get there. So yeah, this study was really designed to be able to address all of these components. And the study participants are really key, as well as those research partnerships that come out of meetings like this. So this is our study. I'll leave this for a second. If you're here and you would like to know more, I also have this in postcard format that I can give to you. You can take away. You can also email me. My email address is here. Or you can reach out to our recruitment center at NIH. Yeah, I'm very thankful to be here, and thank you to all of you.
Thanks so far to all of our speakers, and especially we're incredibly on time. Okay, so we can take a 15-minute break and be back at 10:45, and what we have to stay on schedule too because of the people from Europe who are coming up to speak next, and they'll dial in hopefully successfully. Thank you. So I think we will get started again. And I think our next speaker is indeed on remotely, and will be able to share her slides with us. And we're very fortunate to have Helena Karlstrom, who's head and research group leader at the Division of Neurogeriatrics Department of Neurobiology at the Karolinska. And she's done some very interesting and very recent, I think, intriguing work looking at Notch3 biology, and importantly, developing both passive and active immunization of Notch3, that is developing antibody and perhaps inoculation for it. And this has been published recently, and I know they've gotten a grant and they're sharing their project with the Leiden group, which is coming up next. So we're very interested in getting an update and seeing where this is all going, and I'm privileged to have her talk to us today. So let's see if we can make the miracle here. Hello, Helena.
Hi.
It's
Hi,
all
everyone.
yours.
Thank you so much for the introduction, and thank you also for the opportunity to let me to be part of this really interesting conference, and also to hear about what is going on in the States, and also for the patient point of view. I really have learned a lot during this talk. So I will try to share my screen and see if I can get out my presentation. So, do you see this? Is this okay for you?
Yes.
Perfect. Thank you so much. So, okay. So I will just maybe start to say that I'm coming from the basic research field. I'm not a physician, so I've been working a lot with the cell and animal models, but also, of course, with some patient material, but I'm not seeing the patients like many of you do. But I have close, of course, connections and collaborations with clinicians, both within Stockholm and Sweden, and also in other places in Europe. So this is, I think, a bit unnecessary to start with, that the CureCadasil is the most common form of small vessel disease caused by Notch3. We all know variants which are expressed on arteries, mainly then the vascular smooth muscle cells, and capillaries, the pericytes. And it affects approximately 5 to 100,000 individuals, but we also know that it's most likely underdiagnosed. And most importantly, that we don't have any cure for the disease. So just to give you some bit of a very schematic view of the Notch3 protein. It's a protein which is very important for the blood vessels to work properly, both for the contraction and the dilation. And in it, when we have CureCadasil mutation, it often leads to misfolding of the protein. It starts to aggregate and also recruiting other proteins outside the vessel wall and also within the vessel wall. and we can then find these hallmarks of the disease, which are GOMS, and we can also view them on MRI, like white matter hyperintensities, and so forth. So also to give you a bit of a molecular background, and I try to keep this very simple. So Notch3, it's a receptor, which means that it's situated on one cell, which is the vascular smooth muscle cell or the pericyte. And for the normal function, Notch3 needs to find its partner, like a ligand, which is expressed on another cell. And thereby Notch3 is activated. And this activation leads to a lot of different events. It's cleavage of the protein. One part is going into the cell, showing here, and it activates downstream target genes. So that is the Notch3 normal function. It leads to activation of other genes. When that has happened, this part of Notch3, the extracellular domain, is then taken up by the ligand-expressing cell, and then it's degraded in the lysosome, which is a compartment in this cell, degrading a lot of proteins normally. So this is how it works in the normal setting. However, when we have a Notch3 mutation, that this part, that the ligand and the extracellular domain is taking up and degraded, it's not happening. So there we then find that this extracellular domain starts to aggregate and leading to this aggregation around the vessel wall. And this is also a schematic figure how the Notch3 protein can look like. We have this big extracellular domain, which is starting to aggregate, and these are the intracellular domain. And these bars, they are the mutation which has been identified, and there are around 300 mutations discovered so far that leads to CureCadasil. And often these mutations are located in different regions, and in the first very early part of the protein, we can find the mutations here, and we call that as a hotspot region. So how can we then visualize this? Well, we can look at the MRI, of course, with the white matter hyperintensities, but on a more molecular level, we can also stain the tissue, either from the skin biopsies or from the brain tissue, with different antibodies. And we can visualize Notch3 aggregation and accumulation around the vessels. And this is just to show you here. We have a vessel here, and we can see this dark staining here. That's the sign of the Notch3 accumulation. We can also use immunofluorescence, and maybe this is a bit hard to see, but we can then label the antibodies with the different fluorophores. In this case, it's a red fluorophore, which then detects the Notch3 aggregates. And this is just to which has been a bit computerized to see that we can find these small red dots, which is then the Notch3 aggregates around the vessel wall. And of course, on electron microscopy, with a very high resolution type of microscopy, we can find this Notch3 and GOMS, granular osmophilic material, around the vascular smooth muscle cells. And this is indicated here. Around just in the on the borderline of on the membrane of the cell. So our hypothesis is quite straightforward, that CureCadasil is caused by Notch3 extracellular domain aggregation, and clearance of these aggregates will rescue to vasculature to its normal function and improve or abolish the symptoms for the CureCadasil patients. So we want then to investigate if immunotherapy can be used to clear away these aggregates. So there are different forms of immunization. We have something called active immunization, which means that we immunize, in this case, our CureCadasil mouse model with this Notch3 aggregates. And the CureCadasil mouse then evokes an immune system towards these aggregates. And this immune system then recruits or targets the Notch3 aggregates around the vessel wall, and also recruits its own immune system to come and clear away the aggregates. Another way to perform immunization is something called passive immunization. Then we actually generate the antibody in the lab, which are targeting the Notch3 aggregates. Then we can immunize the mice with this antibody instead. But in the same way, the antibody is binding to the Notch3 aggregates and then recruit the immune system to clear away the aggregates. So what have we done so far? So we have performed an active immunization, a sort of a preventive study before actually the Notch3 aggregation starts in a mouse model. And this mouse model has been generated by our collaborators, Sasna Lesnik and Julie Rattan, which you also will hear after my talk today. And we have immunized a mouse model which has an onset about 5 months of age. So we wanted to come in before the onset, so we started to do immunization when the mice were 3 months old, so before Notch3 aggregation. And this is just to show you how we actually purify the antigen, which we then immunized. So we aggregated everything in vitro, and then we took all these aggregates and immunized in the mouse model. We continued for 4 months. We took some blood during the time, and eventually we took down the mice and we looked into the brain and to the blood and other organs to see the effect of the active immunization. So first of all, we were, of course, very interested to see if the mice actually evoked an immune response towards these Notch3 aggregates. And if you can see here with the red lines, these are the vaccinated mice, and the blue lines are the mice which didn't get anything except for pure buffer. And after 1 month, we saw that the immune response started to happen in the vaccinated mice, and it was quite clear immune response after 2 months. But after 4 months, when we sacrificed the mice, we saw that all the vaccinated mice had a really high immune response. So we also looked into the blood to see if we could detect any of this Notch3 extracellular domain, which are aggregated around the vessels. But could you also detect that in the blood? So we first looked in the mice, normal CureCadasil mice when they were 3 months old without being part of the vaccination, and also when they were 7 months old. And we observed an increase from 3 to 7. But after immunization, we observed a decrease in the vaccinated mice, meaning that suggesting that this Notch3 ECD protein could also be visualized and detected in the blood, and could maybe also serve as a useful biomarker. Then, of course, we were really interested to see if something had happened in the brain, and especially around the vessels with the Notch3 extracellular domain aggregates. And maybe you can appreciate that we have some green staining here in the middle. And this is a 3 months old CureCadasil before, which has not been immunized. And this is a 7 month old in the middle, and this is an 18 month old mouse in the bottom here. And we observed an increase around the vessel wall from 3 up to 18 months. So it was an increase in the number of deposits, And also we measured the size of the deposits, which was also increased from 3 to 7 up to 18 months. So what happened then when we have immunized the mice with this Notch3 aggregates? So just to show you these important numbers here, which are circled in. So the sham, which means that they didn't have any Notch3 aggregates immunized, and the vaccinated here, we saw that both the number and also the size of the deposit were decreased after immunization. So this was, of course, very, very exciting and very intriguing for us to see this. But of course, when it comes to any treatment, we have to be aware of side effects. And one way to look at side effects for the vasculature is to look into the retina, because the retina's vasculature is mimicking the brain vasculature. So first of all here, you can see a normal wild-type mouse with a retina, a very nice vascular tree here in red. And beside that, we have a CureCadasil mouse, which has not been immunized, but the same age, and it looked the same. Then we looked into the sham vaccinated mice, which didn't have any Notch3 aggregates injected. There were no signs of any vascular degeneration, and nor in the vaccinated mice. And this image below here is just to show you how it can look like if you have degeneration of the vascular smooth muscle cells, which comes from this Notch3 knockout mouse model, which have a lot of gaps here in the vascular tree. So we didn't see any signs of vascular smooth muscle cell loss after vaccination, which was really good. We also looked into other organs like kidney, and we didn't see any differences in morphology between the sham and the vaccinated mice. And we also looked for cell death with an apoptosis marker. And there were no difference between the sham and vaccinated mice either. We looked in the blood for inflammation markers. We didn't see any differences, or we didn't see any differences when we looked for neurofilament light chain protein in the blood, which is a sign of neurodegeneration if you have an increased level of this one. And we didn't see any differences after vaccination. So this is just to summarize what we have done and what we have shown with this active immunization in this quite mild CureCadasil mouse model, which has quite a late onset of Notch3 aggregation. And we could see that the vaccination gave rise to a high antibody response in the mice. And also it reduces Notch3 aggregates in the brain vessel and also in the blood. And what I didn't show you here, but we could also see that the microglia, which is like the cleaning cell of the brain, it's involved in the clearing of the aggregates. And we could also see that the vaccination is safe and tolerable in this preclinical mouse model. So this was published last year, and this was also together with Saskia and Julie, and also other people here involved in the study. What we want to do now is to perform a new active immunization, 2.0. But now we want to use another mouse model, which is a more severe mouse model in terms of Notch3 aggregation. It starts already before actually 2 months of age. But what we want to do here is to immunize the mice when they are 3 months in order to see if active immunization also can work when the Notch3 aggregation already has started. So we call it as a therapeutic treatment. The other one was a preventive treatment. What we also have in pipeline, but we haven't started yet with this immunization, is to do a passive immunization, which I talked about previously, is to actually generate an antibody towards these Notch3 aggregates, which we also want to give this more severe CureCadasil mouse model to see if we can reduce or halt the ongoing pathology. So this is the schedule, and for the active immunization, we started now in May, so a bit 1 month into this treatment, and we will continue until February. So this is a long experiment, and we will give them this aggregates, but what we also have done here is to refine the aggregates a bit. And this is to show you here how we did it. First time is that we took all the aggregates here, total aggregated, which means that we also have this very simple monomeric form. However, we believe that this is maybe not so good to have this form, since we don't want to disturb the normal function of Notch3. It's very important to keep that intact. So what we have done in the lab is to actually separate the monomers from the other higher forms of the protein aggregates. And this is what we then have immunized the mice with. So we have just given them 2 boosters, and we will of course collect blood during the time. We will make some half time to take out some mice to see how the effect is, and also to do some analysis on these, but continue with the rest of the mice until they are 12 months of age. And we also have started to generate antibodies towards these Notch3 aggregates as well. And also here, we will use only the higher forms of the aggregates and not this simple monomeric form. And this is a technique which we want to use an antibody phage library that could have thousands and thousands of different sort of antibodies, which you can screen in multiplex format. And then we can select out the antibodies which are binding only the aggregates and not to the monomeric form. And this is something which is ongoing right now. So we are in the selection process here, and hopefully during the fall we will find a suitable antibody which we then can start to immunize in the mouse model. So what happens after all this? From mice to patients, what is needed? Well, as I said, we are working with mice, and there is, of course, a lot of things to do before we can come into patients. We want to do some preclinical development, that's called the safety aspects, before we can head into clinical trials, both for this active immunization, but also for this passive immunization with the antibody. And this is just a bit of an overview of the schedule. And we are now here in the middle of 2024. We have performed the first active vaccination preventive study. We are now started to do the active vaccination, the therapeutic study. We are generating the antibodies, which hopefully we can start to do passive vaccination, maybe late or early 2027. So this is just to thank all the people, of course, which has been involved in the work, from my present and past members in the group, all my collaborators, and also for all the funding, and especially CureCadasil, to give me the opportunity to continue with this work together with them. European Joint Programme Rare Disease and Dutch Brain Foundation. And I thank you for your attention.
So thank you very much for your excellent presentation and giving up time on Saturday to present to us. And hopefully you'll be able to stay around through our panel that will be coming up and field any questions and whatever coming from the audience at that time. But thank you.
Yes, thank you.
And now we'll switch over, I think. So you already had an introduction from Helena for Julie Rutten, who's going to speak on patient stratification and biomarker development for Notch3 therapy, targeting therapies. And Julie is well-known to CureCadasil. She's done webinars for us in the past. And she's a researcher for the Dutch Cadasil Research Group and has worked over 10 years with Saskia, who you also heard mention is collaborated with the Stockholm group. And she's actually performed some very seminal work on discovering a high frequency of Notch3 mutations in the public genome databases, as well as discovery of Notch3 mutation position effect on Cadasil, which is very eye-opening and important work going forward. So Julie, hopefully everything works here and you can take it over.
Okay, so thank you, Bert, for the introduction and of course for the invitation to speak here today. So I have to admit, it feels a bit strange to be sitting here alone behind the laptop in a room. But on the other hand, it's great to be able to be part of this meeting in this way and to hear about all the engagement of the patients, which is really inspiring, and also the work which is now being initiated in the United States. So I will also share my screen so that you can see my presentation, hopefully. So can you see the slides?
Yes.
Okay, thank you. Okay, so yeah, I'm going to follow up on the talk from Helena about the Notch3 immunization therapy. So as you already mentioned, we are now in a joint project, so with Karolinska and the LUMC, to further develop this potential treatment for Cadasil, where we in the LUMC are mostly focused on the clinical part of the project, which is on working towards clinical trial readiness for Cadasil, with a focus on patient stratification and biomarker development. So for today, I would like to start with a short history about Cadasil in the Netherlands. So the first Dutch Cadasil patients were diagnosed in 1998, which was 2 years after Notch3 was identified as the causative gene for Cadasil. And at that time, Saskia Lesnick-Overstein, who is still the PI of our group, she was doing her PhD on this very novel disorder at that time. And I know stories about her having to travel by train throughout the Netherlands to speak to neurologists about this rare novel disorder, and selecting patients with early onset stroke and dementia to be tested for Notch3. So at that time, we had a publication in Neurology, which was basically describing that we had again 3 new mutations in Dutch patients, which was very special at that time. So now the situation has changed quite dramatically. So we have identified starting from 7 families in 1998. Now in 2024, we have over 250 Cadasil families, which include a multitude of patients. And within the Netherlands alone, we know of 50 different Notch3 variants that cause the disease in our country of 17 million. The LUMC is nowadays the national government-endorsed Cadasil Expertise Center. So we see Cadasil patients from throughout the Netherlands in our multidisciplinary Cadasil outpatient clinic, where all the relevant specialties are involved. And our hospital, which you can see here in a picture, is connected via a bridge to our research building, where we have the Notch3 diagnostic lab, where we do the DNA testing for all of the Netherlands. and where also the lab part of our research group is situated. The research we do is really translational in nature, so many of the research questions are inspired by the stories that we hear from patients in the clinic. We do large natural history studies in which CureCadasil patients participate. We have a large CureCadasil biobank, which contains brain tissue of patients, which they donated to our research after their demise, but also the tissues and fluid samples that we collect during our natural history studies. And for the more lab-oriented part, we are working a lot on in vitro and in vivo disease modeling. So the mouse model that Helena is now using was generated in our lab. And we also work on vessel-on-chip models, which are made out of cells from CureCadasil patients. And in the end, the ultimate goal of our research, of course, I think as for all of us, is to find some sort of treatment for CureCadasil. So next to all the specialties and researchers we collaborate with, this is really the core at this moment of our research team. So all the people which are really dedicated on working on CureCadasil, with Saskia as the PI of the group. We have postdocs working on biomarker development. Mark Villacuenca as a researcher who has also been funded by CureCadasil for his CureCadasil iPSC vessel-on-chip model. And we have a number of PhD students who are doing the clinical studies supported by a research technician. So then moving to the actual subject of the talk today, patient stratification and biomarker development. So first, to clarify the terminology a bit, when we talk about patient stratification, in research we often use CureCadasil cohorts, or we refer to a group of CureCadasil patients, which of course share a common characteristic. They have CureCadasil or the genetic predisposition for CureCadasil, but actually, in fact, they're of course not a homogeneous group at all. They are individuals of different sex, different ethnicity, different age, different disease stage, and also different underlying Notch3 pathogenic variants. So with patient stratification, we try to better specify the subgroups which are present in your cohort, and to also better focus your therapy development or your biomarker development on a specific subgroup. And then the biomarker development relates to the fact that if you want to do a clinical trial in the future, you need to have some sort of measure to assess whether your therapy indeed has a beneficial effect on the patient. So this can be all kinds of readouts, including MRI or serum, anything which is sensitive to measuring a change within a relatively short time frame and which is relevant for your disorder. So the fact that CureCadasil is a genetic disorder in terms of therapy development gives an important advantage, which is that you can identify patients, or at least individuals at risk for CureCadasil already years before they have clinical symptoms. The Notch3 variant is present from birth onwards or at conception already. And there are many, many years in which the accumulation of Notch3 protein in the cerebral vasculature takes place before it leads to any clinical symptoms. So that gives a large window of opportunity during which we can intervene with a disease-modifying therapy, such as the Notch3 immunization therapy. This also means that you will have to be able to stratify your patients already in the pre-manifest stage, for example, based on genotype, and that you will need a biomarker which is already sensitive to these changes in the pre-manifest stages. So to start with this patient stratification based on genotype. So one very important discovery which really changed the way we look at Notch3 and CureCadasil was the fact that we found in 2016 that a characteristic type of Notch3 variants which cause CureCadasil, which are the variants that lead to a cysteine amino acid change in the Notch3 protein, occur at a very high frequency worldwide. So we looked in genetic population databases, which at that time just became available. And if you think back, at that time, we estimated CureCadasil prevalence at about 1 in 30,000. We already thought, well, there must be some sort of underestimation because we saw patients being misdiagnosed, are diagnosed with different disorders, but when we discovered that it was 1 in 300, so 100-fold higher than what we had expected based on CureCadasil prevalence estimates, that was really a very unexpected finding and led to lots of questions, which we are actually still trying to answer in our research. What we did discover is that these Notch3 cysteine variants, or these CureCadasil type Notch3 variants, are associated with a very broad disease spectrum, which we now refer to as the Notch3 associated small vessel disease spectrum. And this ranges from CureCadasil at a very severe end to much more frequently a milder and late onset small vessel disease. So for example, an individual in their 70s experiencing their first TIA and being diagnosed with mild cognitive impairment at age 76. But we even see individuals in these large population databases which have a completely normal brain MRI still at age 70. And of course, a very crucial question is, why are certain individuals with Notch3 cyst variants at risk for severe disease, whereas others have a much milder disease course, because this can give important clues for a therapeutic intervention. So this high population frequency of Notch3 cyst variants also gives challenges for therapy development, because we know that approximately 30 million individuals worldwide have such a variant, but how are we now going to identify those individuals who are at risk for the severe disease? So those individuals with the highest unmet medical need who would benefit most from a disease-modifying therapy. And then second is this very variable disease severity between individuals, because if the disease severity is so variable, how are we then going to measure a therapeutic effect in the clinical trial and discriminate that just from normal disease variability? So one of the things we discovered is that the position of the Notch3 variant matters a lot when you speak about the risk of developing severe CureCadasil. So this is a schematic representation, slightly different from, but representing the same as what Helena showed. So the exons encoding the Notch3 protein, where this schematic representation mostly shows the 34 EGFR domains of the Notch3 protein. So these are modular protein subunits, which each contains 6 cysteine amino acids. And if one cysteine is changed because of the Notch3 variant, so it's changed to another amino acid, or a given amino acid is changed towards a cysteine, this in the end leads to the protein aggregation. And CureCadasil type Notch3 variants can be found in any of these 34 EGFR domains. What we did was we compared the cysteine variants which we find in CureCadasil cohorts to those we identify in the population. So I'll walk you through this figure. What you can see here on the x-axis are the 34 EGFR domains below. And then in the reddish colors, you can see the distribution of Notch3 variants which we encounter in CureCadasil cohorts from both Europe and Asia. And below, in the greenish colors, are the Notch3 cyst variants which we identify in population databases such as UK Biobank or NOMAD. I think what you can immediately see is that there's a big difference in distribution. So for example, in the first 6 EGFR domains, those variants are almost exclusively seen in CureCadasil cohorts, are very rare in the population. Whereas, for example, in EGFR domain 31, many variants are found in the general population, and they are very rare in CureCadasil. So based on these differences in Notch3 variant distribution, we divided the EGFR domains into high, moderate, and low risk for developing CureCadasil. So the high risk EGFR domains are the variants in there almost always cause CureCadasil. The disease course can still be variable, but individuals with a high risk EGFR domains almost always at some point in life get the clinical symptoms of CureCadasil. The moderate risk EGFR domains are a bit in between with a very variable disease course, and then the low risk EGFR domains, those are the variants which are found in the population quite frequently. And interestingly, CureCadasil patients, we do see CureCadasil patients in the clinic with a low risk variant, but it's rare. Those are the exceptions to the rule. So it can happen, but most individuals with a low risk variant get a much milder disease course. So we analyzed a number of outcomes based on our stratification into high, moderate, and low risk. So this Kaplan-Meier curve shows you that both within CureCadasil cohorts as well as in population databases, high-risk variants are associated with the highest, earliest onset of stroke compared to moderate risk and low-risk variants. And we can see similar differences when we look at brain MRI markers. So the white matter hyperintensity volume, a measure for microstructural brain damage, but also the number of lesions on the brain MRI. And this is here the comparison between high risk and moderate risk. We see that for each of these MRI markers, the burden is higher in individuals with a high risk variant compared to a moderate risk variant. So one of the questions we then had was, well, why does the position of the Notch3 variant matter? Why would certain variants cause more severe disease? And we actually found at least one of the clues. Because when we looked in skin biopsies of patients in our CureCadasil biobank, we found that those high-risk variants are associated with a higher Notch3 protein aggregation load in the vasculature compared to moderate risk and low-risk EGFR patients. So I'm just showing representative pictures here, but you can trust me that we've looked at this in 200 patients and we found very, very consistent effects. So apparently those Notch3 cis variants in high-risk EGFR domains lead to a Notch3 protein with a higher propensity to aggregate than the Notch3 variants which are located elsewhere. So then moving on to the biomarker development, and I think that an important challenge here is the time frame that we have to work in, because we've been speaking to many biopharmaceutical companies, and one prerequisite when they want to initiate a clinical trial is that they want to be able to determine whether your therapy actually has a therapeutic effect within the short time frame of 2 years. So to search for these biomarkers, we have set up a large prospective follow-up study, which we call the DVNA study. This stands for Disease Variability in Notch3 Associated Small Vessel Disease. And in this study, we had included 200 CureCadasil patients or relatives of a CureCadasil patient who have been tested positive for the familial Notch3 variant. And we've seen all these patients at baseline, which was between 2019 and 2020. And then 2 years later, all these individuals were re-invited for the follow-up assessments in the DVNAS-2 study. From all patients, we obtained medical history. We did an extensive brain MRI protocol, a neuropsychological assessment. We took blood and skin biopsy. And at the second time point, we have also collected cerebrospinal fluids from a subset of patients. And we've done retinal vessel imaging. So then moving on to the first results of this DVNAS-2 follow-up study, which have recently been published in Neurology, where we found that it makes a difference. The disease progression differs between individuals with a high-risk variant compared to those with a moderate risk Notch3 variant. So here you can see examples of 2 outcomes that we looked at, so 2-year changes. Under A on the left-hand side is the mean skeletonized mean specificity, which is the marker for microstructural brain damage. It's a relatively early marker for CureCadasil. And what we found is that this measure is sensitive to measuring changes within a 2-year time frame, even in young individuals, even those are the pre-manifest individuals without any clinical symptoms. Also there we can already measure changes in mean skeletonized mean diffusivity in 2 years' time. And we can do this both in the high-risk patients as well as in the moderate risk Notch3 patients, although the high-risk Notch3 patients do increase faster than the moderate risk. For the normalized legume volume, we found that this could be a potential biomarker for the high-risk individuals because those were the patients that showed a significant progression in normalized legume volume in 2 years' time. The moderate risk Notch3 variant patients did not show this increase in 2 years' time. So this brings it back to the patient stratification and the importance for therapy development. So if you have an individual with a moderate risk Notch3 variant, you might choose or prefer MSMD if you want to use an MRI biomarker as an outcome measure rather than normalized vacuum volume. So to conclude, we can now, based on genotype, stratify patients into having a high, a moderate, or a low risk for developing a classical, a severe CureCadasil course. I have to say I did not go into that here, but there are, of course, other modifiers such as male sex, cardiovascular risk factor profile, and also still a lot of disease modifiers which are unknown. And those are also the focus of our current studies, to find these other modifiers. We have shown in our 2-year follow-up study that MRI biomarkers are sensitive for measuring 2-year disease progression in CureCadasil, and that we found that there is a difference in disease progression between patients with moderate and high-risk Notch3 variants. So that brings me to the ongoing studies we are now doing on patient stratification and biomarker development. So together with Helena, we are now working on a 4-year project funded by the Dutch Brain Foundation on Notch3 immunotherapy for CureCadasil, where we are testing other biomarkers from our DIVANAS 1 and 2 studies. So also the serum biomarkers, for example, but we're also looking in the skin biopsies. And for patient stratification, one of our postdocs, Guido Gravenstein, is currently working in an international project involving CureCadasil centers from throughout the world, patients from all kinds of different ethnicities, to develop an international Notch3 disease staging scale. So next to classification on genotype, also classify patients in a consistent way based on their disease severity. So with that, I would like to finish. To thank you again for giving me this opportunity to present our research, and thanks to all our collaborators. And I would like to hear, as we are also speaking to the patient organization, also mention our Dutch-Belgian patient organization, which is called Platform CureCadasil. They organize meetings for the Dutch and Belgian CureCadasil patients yearly. So we had the last meeting in April in our center with over 100 CureCadasil patients attending. I think they would be very interested in also connecting with you to see what they can also learn from you, and vice versa. So thank you for your attention.
Thank you very much. And I think we're going to try to now transition to our panel. But as I understand that the last 2 speakers we can have on the screen, and they can participate also. And then if we can get our other speakers up front, we might need a couple more chairs. So Pedro and I will float around. If people have questions or comments, we'll bring you a microphone, and you can for sure express them to any of the panel members or specific panel members. I can start with one, because Julie had promised that she would comment on their ASO, and that they have been looking at, I know, for a number of years. But I think is it on hold now, or what has happened, and what specifically kind of complications have you run into? We'd love to hear that.
Yeah, thanks, Bert. So we had indeed, I think in 2016, we published on an ASO therapy development for CureCadasil. So this was an exon skipping approach, where we try to remove the mutated exons from the Notch3 mRNA in order to generate a protein which is no longer has the propensity to aggregate, but still retains normal signaling function. So what we have running up against, so we have been continuing working on this all the time, actually, also together with multiple biotech companies who have been helping us. The major challenge, there are actually two challenges. One is that in order to bring such a therapy to the clinic, you would need one single ASO efficient enough to skip your target exon. And exon 4, which is the exon which harbors the most of the high-risk variants, so would be the first one to target, is very difficult to skip. And we are only succeeding in that when we use 2 ASOs, which for biopharma companies is unacceptable. So the skipping efficiency is one big challenge, and the other challenge is getting the ASOs into the target organ, so into the cerebrovasculature. So we've done many mouse experiments where we can get the ASOs very efficiently into all kinds of organs, but not so efficiently into the cerebrovasculature. So these are the two hurdles which we are now running up against. So what we have to hope for at some point is novel ASO chemistries. So there are novel ASO chemistries coming onto available all the time. So we hope that at some point there will be something with which we are able to efficiently skip exon 4. And then the delivery is the second thing we will have to improve to bring this further.
Great, thank you. So please, any questions, comments? If you want, you can identify
Julie,
yourself or not.
Hi, I'm Barbara Hunt. I was the founder of CureCadasil, and my husband has Cadasil. I have 4 children and 7 grandchildren who are potential patients, so I've remained very active, even though my husband is at that late stage of Cadasil. I am so happy to see these 3 studies and to feel that maybe I had a part in getting you here. But I'm a little confused about exactly what each of your studies are doing. I'm a little bit clear on yours, Fanny, but on the consortium study and the NIH, how do they overlap? What's the differences? Should patients be involved in all 3 studies? Is it just going to be repetition? You know, do they, because they need to go through MRIs and blood work and cognitive assessments in order to do that? So
Yeah, so at least for the NIH studies, we are doing natural history of Cadasil. There are tests that do overlap with the other studies. Dr. Elahi and I have talked, and we don't have as much overlap. I don't think. The brain MRI for us right now would have to be done at our clinical center just because we have to control a number of things. But there may be tools that allow us to change that in the future. We are not just doing the clinical studies. We're also collecting cells from the patients and doing the iPSCs and the stem cells and doing the vascular models. So that is one component that probably overlaps with Dr. Elahi, but not necessarily with the Wisconsin study. And then for neurocognitive assessment, there could be some kind of learning effect from repeating those over and over. So when we looked at some of these tests, there's not as much overlap as we initially thought. And there are different tests that can be administered to assess the same domains. So there are things we can do there, as well as some separation from maybe you don't come like 2 months after you've had one of those tests, but you do it a year later or something like that. For ours, I guess the biggest thing is we're interested in the very long-term follow-up. 9 years is really a commitment for us, but also for you guys, right? That's 9 years of you planning a visit to the clinical center. And then I don't know of many other studies that are specifically studying the vasculature. Of course, there's everything that's happening in the brain, but we're interested in seeing other parts of the body where maybe Notch3 is having an effect.
So I think this is a really key question, Barbara. And I think we ought to be pushed all together to figure out a way to collect a minimum set of data that can be used that year for that patient in all the different studies. And so how I conceptualize it is, in the consortium, you have the core and then you have ancillary. And so the core, we should come to an agreement of this should be the sequence of MRI, like basic, right? And so if that patient had it in 2022 in one of the studies, all the studies would be able to access that MRI and wouldn't need to scan them again. Blood is something that we do need to collect, but even so, we could consider a central bank and people could use it. As I was looking at the map of the countries, we're a continent. So it's sort of normal to have multiple studies in the US. France, almost everyone is going to sort of a center of excellence in Netherlands and Sweden, and we are really large. So to lessen the burden of travel, there ought to be multiple studies, and it's a headache of red tape. So I don't think there's a lack of willingness because we've had this conversation, but because there's so much work to be done to be able to actually effectively share, it hasn't happened. But I think we do need to make that happen this year. So I think we need to figure out how to share. And a central repository would be an ideal way to do that, uploading to a cloud-based system.
So one of the things that I'm trying, oh, thank you. One of the things that I'm trying to, I guess, understand is it beneficial for patients to enroll in all three studies? You know, and I think it's asking a lot of patients to have MRIs done 3 times because that's like a lot of radiation and stuff.
Yeah.
And the cognitive tests, which is
Right.
very involved and exhausting for patients with CureCadasil. So, yeah, if there was some way that you
Yeah.
could all work
So-
together and share and I don't know, it's just,
So
and I understand you're trying to do it.
I think one of the things that we did for a long time until now was not to get an MRI and to ask people to give us their clinical scans, and worked on collaborations with others who had developed AI machine learning algorithms that could use noisy data. Because one of the limitations if you don't acquire your own MRI is that that MRI is going to come from multiple different kinds of scanners and multiple different kinds of protocol. But we didn't want to ask to scan everyone because we thought that would be the biggest limitation. We are starting to do a specific kind of scan that I know you guys are also doing because it's a very specific sequence. It's not clinical. But we, in our study, could not scan for the most part all the scans that they are getting. We would just need to have access to that if we're not going to acquire. So that problem could be solved. I think so. And maybe that's a deliverable from this, and you could hold us accountable next year if we've achieved it. That's a good amount of 12 months to work on that. The other thing that we can't not do in our study is the blood. So one alternative, which Harrison is leading in the group, is remote phlebotomy so that people don't need to travel. And we will, this year, in the next few months, analyze all of that to see whether it's compatible with the in-person phlebotomy, whether the quality is good enough. But I'm hoping that it is, and so that would lessen the burden of yet another travel for a blood draw. The component of immune system, which hasn't been mentioned today, except for therapeutic of immunotherapy, and Helena and I actually had a meeting this past week, and hopefully we'll continue to discuss work, is that we are getting signal that the immune system is not 100% normal. And as such, we do need to collect immune cells from the blood that can't be done remotely. So if we can implement that component of our study in both these studies, I know you guys are doing, then again, the key component of each study could be helped by the other studies, and that would again lessen the burden. So I'm happy to put down in paper what is essential and non-redundant. And if you guys could do the same, that may be a very good starting point to see how could we lessen travels, CO2 generation in the world, and burden on the participants.
For us, it's really the way the study was designed, right? So the MRI, the brain MRI, and that follow-up is actually a primary outcome of the study. So it's very difficult to do away with it. And this is human subject research. So RRB is the red tape. But there's for sure always a willingness. And a lot of what we do is actually clinical. So all the clinical scans are something that we can definitely share with a physician, right? It might be more difficult if it is like a PhD PI, because there wouldn't be a clinical reason to share, but that's already something that we can do. It's really the research portion that's very difficult to figure out how to share right now. And it's a conversation that's ongoing.
I just wanted to add two things. I think you've been in more than one trial. Maybe you can comment on how it works from your perspective. And the other thing is there are so many different approaches we can take to trying to understand these disease so that we can treat it. And I think individuals should feel, whatever study works out for them is most convenient. It's better to have a multiplicity of people looking into it than a paucity or only like one mindset. So I think we certainly should be sharing data. But I also don't conceptualize multiple different groups working on it as necessarily a negative, if that makes sense. And of course, people individually shouldn't feel obligated to join more than one study. But I just want.
Yeah, for myself, it hasn't been challenging. So I participated in the consortium, and I live in that area, so that wasn't challenging travel wise. But and then in Dr. Elahi's study, I participated in that virtually. So I had a nurse come to my house. We handled it. It was very easy, very quick. I got to work just right on time, and it was very convenient and easy. But yeah, of course, there is overlap. Several blood draws and such. For myself, I don't consider myself very. I'm healthy. I'm not super limited. So for myself, participation in several studies has not been challenging, but I certainly can understand that burden otherwise. But overall, it's been fine for me.
I have been told that there is no evidence that multiple MRIs is detrimental to one's health or brain function.
Yes.
Oh yeah, it's certainly inconvenience is another factor, but
Ours is quite long as well, but they don't have radiation, and so they don't cause any accumulated harm, which is good. But they are still psychologically painful to endure. So, and unfortunately, there are many exciting new techniques in MRI that we keep wanting to add on and acquire. So, I think it's smart maybe for the consortium study, you can agree to do the additional time for those MR studies or not. So we may be able to break down the amount of MR you're willing to sign up for.
I'm not sure if everyone can still hear me, but this is Courtney. I am currently involved in all three studies. So, there is definitely an element of, it can be stressful. It's obviously a big time commitment. And I definitely understand the sentiment of, it would be great to have more of an overlap and share some of that data. But I do feel like I've been able to take slightly different takeaways from all the different experiences. Each researcher, each doctor has slightly different things that they're looking at, slightly different information and perspectives that they are able to provide. So I think if folks have the time and the ability to participate in multiple, from my perspective, I think it's been a good experience.
I have a question for the public. So if you were to be given your MRI on a disc from this, would that be burdensome? Would you like to have a copy of your scan, or you would rather not know, not see?
I think it's helpful personally.
So that may be a possibility if we don't come up with a sophisticated way of sharing data. We have our IRB approval to give you the scan if you wanted. So we would be able to burn it on a CD, give it to you, and then you could take it to another study. That could only happen for the core elements. And we haven't done it for the neuropsych. We haven't actually inquired, but we could inquire and see. You know, these are all research grade. The MRI isn't. That's clinical, actually. Same scanner, same protocol as a clinical scan. But the cognitive testing is not done by a neuropsychologist. So we would need to see whether we can get approval. But we could work on giving the individual their data. And that individual would be open to sharing it with who you wanted to share.
Also do this, the CD, the burn CD.
We're just working on it since most of our protocols are research protocols, but they have been working on a process to make it interpretable as well.
Sorry. Just really quick, Tom, I forgot to mention, was in NIH 2017, 2019. It was fantastic. It was just such an amazing, positive experience. Thank you guys so much. But I wanted to say, you know, every year we would bring all of Tom's data and any scans he would have, and then in return, which I thought was so fantastic, NIH would give us his reports, his scans. Dr. Toro taught me how to look at the MRI. This, you know, this is this and that is that. It was so incredibly helpful. And then what we did in turn was we gave it to the neurologist and his primary care physician. Everybody was on the same page. And if there were questions, you know, NIH was so amazing to, you know, constantly, I felt for like a year, okay, what about this? What about this? And okay, well, this is this, and you know, that's that. But so our questions were answered, and I felt really confident that Tom was just getting the best care he could because everyone was on the same page. So I thought that was really incredibly helpful.
So both, excuse me, both Julie and Helena have chat questions. I don't know if you can see those or have Pedro read them.
Okay, yes, sorry, I can see it. Also engaged in your discussion, so I didn't look at the chat. Do you want me to answer the question here or in the chat?
Yes, please.
Okay, so the question I think was, sorry, why not use first model to test treating in symptomatic phase? Okay, so you mean, yeah, so we started with a mouse model with active immunization in a mouse model which didn't have NOT3 aggregation ongoing. And the reason for that was that usually active immunization you actually use as a preventive study normally if we think about active immunization for viruses and so forth. And so we wanted to do this just to see if we have any effect at all. And since that also was quite a short study, we decided to go for that. And since we also now saw an effect, we of course want to see if we can do the same thing in a model which has an ongoing pathology. And I know that the time is a bit longer, but we will take some after the same time of period as for the first active immunization to see if we have an effect. So I think both are valuable knowledge for us to see if actually active immunization can work for both pre-symptomatic and symptomatic patients. And we're coming to passive immunization. we don't know at all. We know from other neurodegenerative disorders like Alzheimer's disease that antibody treatment is working better in the early stage of the disease. They haven't given any antibodies to pre-symptomatic patients, so that we don't really know, because we don't also know when the patients will actually have the disease there, if we don't use the familial Alzheimer's disease patients, but that has not been done. So we will just have to wait and see how that goes with the PASS immunization later on.
Maybe I can answer another question from the chat from Michael Kushwins about what's the effect of removing exon 4. So this relates to the ASO therapy I answered the question about before. So if you remove exon 4, then you remain in frame, and on the protein level, you remove EGFR domains 3 and 4, and a part of EGFR 2 and 5, and what you get is what we call an EGFR fusion domain. And that fusion domain, so that's the remaining parts of those other of EGFR domains 2 and 5, they together, this fusion domain has again 6 cysteines. So that's the canonical number of cysteines, which you also have in wild type EGFR. So that's the idea behind it.
I guess, Julie, my follow-up to that was, what happens functionally though? So does the protein still function in the normal way in vivo and in vitro?
Well, in vitro, yes. So we've tested that. We've looked at Notch3 signaling activity for at least for proteins from which we excluded both exon 4 and exon 5, which was our initial approach. And those proteins retain normal processing and normal function in vitro. In vivo, we've not been able to address this because the efficiency is still too low. So you have a lot of just wild type Notch3 there, so you can't look specifically at the effect. But in vitro, it looks like the signaling function is retained.
Great. Did you have a question? Oh, okay. Right. If you didn't hear that, Jane was saying that was Michael Geschwin at UCSF, who has been a great clinical investigator in CureCadasil for a number of years.
Thank you. My name is James. I'm here with the CureCadasil Eradication Project, which is another endeavor to try to use data science and AI to deal with all the complexity of CureCadasil. This may be a particularly American question, so forgive me for those who are on the phone or on the conference call. But having gone through the experience of CureCadasil for now 4 generations in my family, one thing I've seen is the struggle that all of the complexity and the diversity of symptoms that arise from CureCadasil, such as, there was fatigue, headaches, brain cloud, sleep, all these things. CureCadasil can really manifest in different ways and in different intensities. In the US, one of the challenges we have is not only just the crazy insurance that people have to deal with when they're fortunate enough to have insurance, but then also the complexity of what if people try to go on disability and get recognized by Social Security and whatnot for that type of problem. Does the panel have any thoughts around the best ways to navigate that sort of challenge that people I know have to deal with themselves individually as people who are trying to go to their job as much as they can, but they're struggling because they're having a bad day or they didn't get a good sleep or whatever? Any thoughts around that would be appreciated. Thank you.
Yeah, that's a great question. And I think people with CureCadasil, or really any disease like this that's a chronic progressive condition, really benefit from having a multidisciplinary clinical team. So of course that means finding a neurologist that's familiar with CureCadasil. but also documenting all these other limitations. So working with a clinical neuropsychologist to have that documented, and the more documentation you can do, the more successful you are with getting things like disability and other resources. So we don't have a multidisciplinary style clinic for CADASIL. We just kind of involve each group separately, but it's a great thought with a focus on particularly this disease process. Do you guys have any?
So I've practiced in a multidisciplinary setting when I was at UCSF and again at Mount Sinai, but the resources are shared with all the patients that are served by that clinic. It's a great model. So there's a social worker, two actually social workers, and then nurse practitioner, nurse, physicians, a neuropsychologist, psychiatrist, et cetera, et cetera. My dream, so this is maybe something that we can make happen, is to have a center of excellence for CADASIL, because it's a disease, as you mentioned, and as our survey is showing, is incredibly multifaceted in ways that it actually impairs that individual's ability to navigate the system, executive function being one of the main, fatigue, executive function. So the disadvantage are getting more disadvantage, meaning that if someone has the resources, and I have very few patients, but who have incredible resources to get a management service to manage their care, they go from one appointment to another, and that person is like pursuing, making sure that all the physicians are talking, all the recommendations are happening, etc. But why can't we fundraise to make that happen for everyone? So having social workers dedicated to navigating the really lengthy and complicated system of getting disability. Unfortunately, the system is broken and this is not funded by someone's insurance. But if we recognize that, I think it's something that we can work towards fixing and have maybe a few centers of excellence. We would love Mount Sinai to be one, such that we don't just do the research, but on the clinical end, it's an end-to-end care. I know patients who are not even that impaired and have trouble navigating the care system because it's really broken. So
I'll add, actually, we recently started working with the social worker team at NIH, which is something that has been growing as well. Like most teams, it started small and has started growing. And we connected with them over a different disease that we are studying, and realized how important their role was. So we now have social workers connecting with our CADASIL patients as well as all of our other patients to be able to help navigate these pretty tricky situations.
on a practice, so I would love to have a center of excellence dedicated at Sinai, multidisciplinary effort for the migraine specialist who only sees, and I think we can make that happen. But short of that, as we're working towards that, maybe we could, some of the fundraising and donations could go towards a centralized social work, just one or two people who we could connect our patients to, and whose only job is to help individuals who get a new diagnosis of CADASIL and what's the ABCD. The other thing that we're doing, so I think that would be fantastic and something to think about. But the other thing that we're starting to do, it's gonna kick off in August maybe, is a peer support group. So short of having someone dedicated, a lot of you and others have gone through this journey. So we want to bring people together so that there's that knowledge of, I've been there, done that, and here's the trick, and this is what could help. So crowd outsourcing the knowledge needed. And be on the lookout for those announcements of peer support group.
I'll just say with Tom, had the stroke, never returned to work, but where he was, I guess, lucky, if you can call it lucky, he worked for a worldwide insurance company. And so for him, everything was in place. But it was really difficult. I mean, he went from short-term to long-term, then retired out. He had worked for the company for 30 years, so he more than had the time to retire out. But when it came to getting Social Security, because that was the thing, if you have long-term disability, it has to be supplemented by Social Security. And that took like 2 years. And we were incredibly lucky to have a company in Massachusetts called the Investigator Group who went to bat for Tom, and they handled all the paperwork. I mean, we had to do our paperwork, but they handled getting the Social Security. And it's still, and he was still denied first go round. So it's really tricky, and it can be really upsetting, so I think that's something that definitely needs to be addressed, is where is the support? Where can people look? If they are their own advocate, where can they turn to in the community where they can get this help to help them along? Okay.
I'm thinking that this problem isn't a great problem in the Netherlands. Yes, there you go. We'll
Or Sweden.
Here's a question from Dr. Ferrand. Dr. Ferrand, you mentioned that your organization acts as a type of expert in repurposing approved drugs for treatment. Is there a class of drugs you're targeting that you think would be effective for treatment? If so, why? And this is from Craig.
We've done the interventional studies for some of the other diseases that we study. There's a great example of a study on an arterial calcification disease, which was something that we identified the gene. We did the in vitro models in the lab to actually be able to figure out the mechanism, but we also used those in vitro and in vivo models to then take it a step further and test all of the drugs, and then created this interventional study that wrapped up and was published recently. For Cadasil, we're not as far along, but I know that there are conversations, and there's a new working group that is thinking about different therapies that we could potentially target. And this is a conversation I just came into this week because Dr. Elahi gave a presentation at NIH on Tuesday, and we connected over this. So I don't have a specific target quite yet, but we are starting to focus in on this.
Shout out to Sheila, Andrea, Barbara, Jane, many names and Craig yourself, actually. So I think we've been thinking about drug repurposing, but you've done an amazing job at pushing this forward faster than potentially. Life is busy and so much to do, and it wasn't the highest priority, but now there is a document that we've gathered, clinical researchers who've expressed interest, clinicians who've expressed interest. So we've formed this working group where we are dividing up the work of just the basic, what are the toxicities? What are the side effects? What are the indications? What would be the outcome? And everyone is absolutely invited to join. Just send me an email if you're interested to join this effort, because it's the groundwork of what's out there. And then I think together we could discuss repurposing. We have in vitro models. There are in vivo models that our European colleagues have, and we also have those models in the States. But the starting point would be just what's out there. And because it's drug repurposing, we don't need animal models necessarily, which is amazing.
Yeah. Go.
Hi, Dr. Rutten. My name is Megan, and I'm with the Cadasil Consortium. So I've never heard the term Notch3 associated small vessel disease spectrum before, but I think that does a good job of encompassing some of these questions about people who, have a Notch3 variant, but don't have the classical Cadasil cases. And I was wondering if you believe that there is kind of future directions in that, kind of expanding towards these folks who don't who may not have necessarily classical CADASIL, but do have this variant and do have signs of vascular changes?
Yeah, I think one important aspect is that, which has also led to much more interest in CADASIL from, for example, funding agencies, is that before we were talking about developing therapies for a very rare genetic disorder, whereas now we know that many people are at risk for some form of Notch 3 associated small vessel disease, which may in the future also benefit for treatment. So I think that's one, that then the importance for public health has become much broader. I think from our perspective, understanding the protective factors as well as the potential aggravating factors, that can give very important clues for therapeutic intervention, because we know that Notch aggregation plays an important role, but then there must be much more that we don't understand yet. So if we can identify, so for example, we are working in the Netherlands in genetic isolates. So we have a genetic isolate of CADASIL. People from a certain village all share the same Notch 3 variant, so their variant position does not play any role, but still we see remarkable variability in disease severity. So we are now comparing the genomes of these individuals with very mild disease to those with very severe disease in order to hopefully find clues as to what, at least genetically, plays a role in the disease variability. But we also, of course, want to look at environmental factors which might be more easily modifiable.
Thank you to everybody who's given us so much additional insight today on this long journey. But Dr. Ulahi, in particular, you mentioned that several papers have been published as a result of the work that you're doing. Are you able to share any of the topics? Are there preliminary findings or new insights we can benefit from learning about?
Yeah. One of the papers looked at an imaging measure. My colleagues may be familiar, enlarged perivascular spaces. That is basically just spaces around the blood vessels that get enlarged, and enlargement is not necessarily abnormal. Actually, teenagers and kids can have it. When I started studying this, my colleagues started sending me scans and saying, like, what do you think about it? So it's actually quite normal, but there's a dynamic component to that. And it may be the aspect of clearance from the brain, where in the morning, if you scan someone, or in the evening, if you scan someone, the size would actually be different because it's the dynamic component of the brain. So we didn't have 2 scans in individuals, but we looked at the relationship of this measure on brain imaging to blood molecular measures. And we did an unbiased, so meaning thousands and thousands of proteins, and found that in individuals with CADASIL, it's associated with inflammation, signatures of immune system inflammation. It's not groundbreaking, I would say, but it's coming, it's congruent. It sort of fits existing literature that use different kinds of methodology. And it suggests that there is a component of inflammation, immune system reaction maybe, or it could be a component, even more causal component we need to understand. But it does suggest that there may be therapeutics against immune system inflammatory response. You know, my words are not very specific because that's why we need to look at the immune cells themselves in order to become more. So that opened up a whole new line of work, and I think potentially therapeutics. The other paper was an approach to discovery, which is applying machine learning, artificial intelligence. And I think that can be done on many different levels. We didn't have a lot of scans. We did it in blood. and again, proteomic measures. And it suggests changes to the blood vessel plasticity, again, inflammation, and some more specific pathways that are emerging. That work for us was, I would say, game changing because we saw things we wouldn't have otherwise. And this is just the start. I was speaking to James who asked the question. I think our methods can be applied across data sets. We had the great fortune of collaborating with Dr. James Meskia, and we say Dr. Joe because no one can pronounce his last name. So they made their data, they made their biospecimen and data available to us. And it's absolutely critical because if we had only looked in our own data set, there's always this sort of question of, is that true? Is it just the patient? So you need, when you do these approaches, you need at least 2 to 3 data sets. And I think as our collaborations expand, I know that we've been discussing this with NIH, and at some point the consortium is going to have data available in blood as well. We would have new biology that could emerge from that. So thanks for that question. Is it really cold in this room, or is it just like my teeth are borderline gonna show? The one that has been published? Yeah. So James Meskia led an effort that was endorsed by the American Heart and Stroke Association that provided guidelines based on the state of affairs. And this was a large collaboration. I don't know if it extended internationally, but certainly nationally, where I was a behavioral neurologist, and I think there may have been others on that paper, stroke doctors, and so the different sort of angles of CureCadasil. And what was incredibly eye-opening for me was that we don't have concrete recommendations that we can give with regards to these are the medications that you should go on, and this is the risk reduction. So scarcity of data was a really big takeaway. And more so, a bigger even gap was for women. So we know actually that women's menopause is sort of this change in vascular function, plasticity, and health. Should people go on hormone replacement therapy or not? What kind of hormone replacement therapy? What are the risks of pregnancy and giving birth? And there was nothing. And that was my section. Initially, I was not very happy. I was like, just because I'm a woman, that doesn't, but then it was the tiniest section. There were only a few papers to read, so we need more data. And these are all modifiable. So I think that's about it, and some other papers in the pipeline.
On the theme of drug repurposing, the GLP-1 agonist manipulations, yeah, I keep reading about randomized trials that are actually going on now with dementia, not specifically CureCadasil, but other causes or all-cause dementia, because it was noted in the original randomized trials that there was reduction in dementia. And there's some reason to believe that could happen because of inflammation and brain inflammation. And so I wonder if anyone has any, and the other reason is that these are miracle drugs. They affect everything, cardiac disease, kidney disease, and why not, in the CNS. So anybody know about these trials or have any information or insights?
It's on the list for the working group. But in Europe, are you guys looking into the GLP-1 inhibitor? No.
Doctor. So there have recently just been studies releasing showing a reduced risk of stroke in people without diabetes. So we've been pushing them in people with diabetes, but I've already had patients with CureCadasil ask me if it would make sense for them to be on them. And the problem is they're not approved for that purpose now, but I think it's a really interesting avenue of research. So. There might be risk being a CureCadasil patient. True, maybe at risk as well. Yeah.
I'm Patty Mussolino, a doctor, stroke neurologist. Gene therapy in much rarer vascular diseases than CureCadasil. That I have a comment and 2 questions for the panel. One is the the consortium that was formed internationally for common data elements, coming back to selecting a series of variables that each study will kind of deposit somewhere. ALS is a very good example, 256 international centers, common data elements deposited, and in fact is a nonprofit that was created to be able to home that registry. It ends up being NIH-funded neurobank, which has all the data. And I just want to say, each investigator kept the locality and the focus of the things that they do really well and beyond. So that's a very important thing. Don't try to get everybody to do the same. Just do what you guys are doing. It's phenomenal what has happened in CureCadasil in the last 10 years. Is incredible. And I think you guys, CureCadasil, had it, and the patient community has the I think the credit here. But it's important that this registry and database starts existing as soon as possible. If you give me a chance 10 years ago, because when you go to the FDA to get a drug approved for your disease, you'll need this database. And it's not gonna be your group, your study, or anything. It's the aggregate of those patients. AL is a very good example, heterogeneous, many mutations, many mechanisms, many scientists, many people working in different parts of the body for the disease. And it's still, each study has common data elements that they deposit, and you also can access who has biomarkers, who has iPS cells, where is the imaging living, all for each subject. So if the patient goes to different studies, you still can find everything that links to that patient in this database with a common unique identifier, which you guys are doing already. So I would say encouragement on moving the harmonization of data to a self-standing, probably non-institutionalized registry or whatever you guys can decide. But this was used now for 3 FDA approvals that failed to get approved in first place without the natural history in ALS. The first one was February 2023, the second one was September 2023, and the third one which just happened. So it's very, very important. The patients are doing the right thing, the scientists are doing the right thing. It's just the last notch of let's get all the data harmonized. So when someone is ready to go for some therapeutics, even the repurposing has been one of the cases, you'll need to compare to best knowledge, and is a rare disease. So I think I just put all the kudos. And my question was, I see the onset of symptoms always 20 and above. I do the very ultra rare things, and they affect more children, vascular genetic conditions, including GACI and PXE, and I do get the cadasils that for whatever reason were found to have cadasil that are less than 18. This is not common, okay? The people most commonly don't get tested, and I think that's correct. But I have patients with severe migraines and a lot of dysautonomia. I want to ask the question, is the onset of symptoms 20, or do we have to go a little bit to more, like I'm asking because this is a survey that we can do like in the community and really dig in and how early potential symptoms happened to people that do have diagnosis of CureCadasil and that older. You go by memory, it's patient reported, but it's very helpful to start moving that needle of, is there something else that you could be potentially targeting as a symptomatic and a meaningful clinical endpoint?
For our cohort of patients, when we ask about the various symptoms they've had in the past, we ask them to estimate the onset, because I think it's a thing that we don't know. So we'll see if we can get any more data about when people report the onset of the various symptoms in at least that cohort.
I mean, I think it's tricky with some of these symptoms, right? Because migraines happen to a lot of people. And so I think you have to look at the individual patients and the history that they're describing to really figure that out. I suspect the onset is a lot earlier, but of course there's a number of ethical issues when you're testing minors for a variant that is adult onset. So that's something that people shouldn't be willing to do because it will affect the way they grow up, right? But yeah, I think it likely is earlier, but difficult to assess.
Because I don't think CureCadasil is ultra rare, we haven't gone there. There are few parents, who have taken things in their own hands because their children were symptomatic. have shown that their children, now it's not high numbers, so it's not 100% sure, but they do have white matter changes, quite early, etc. I'm just waiting for something to be offered therapeutically, even if it's not a miracle drug, to go earlier, because it does have a really big psychosocial impact. And for that reason and that reason alone, even after 18, I'm asked, should my children get tested? And I'm like, as long as you make sure they don't smoke and do cocaine and sleep and then maybe not yet. What's gonna be different about their life? Yeah.
I completely with you. Huntington's has had this conundrum of tested, not tested. In the research, you can still get the blood, get the mutation. The patient chooses not to be reported, and you can run the survey to every single sibling, every single person in the family, and you get very beautiful unbiased stuff. Like I'm saying, and the other way to do it is just adults that have been diagnosed, and you go back into as early as you can with a more wide battery of questions. But there is a way to do it in research without having to disclose and putting the people through trauma.
Are
Maybe
you
I
guys
can
doing that
add.
in Europe?
Yes. So we actually have done a study on the occurrence of CADASIL symptoms in childhood, which has not yet been published, but we've looked into this, and so far we have not seen any childhood onset. And I agree with what I think Fanny Elahi said, that the things like migraine are of course very difficult because they occur so frequently in the population. And what we actually sometimes see is the reverse, that a child with a parent who has CADASIL, so the child has 50% a priori chance, and then any kind of symptom is then a reason to do genetic testing and then saying, okay, so this is the onset of CADASIL, but the child just had 50% a priori chance. So that's not very sensible, and also I think something we have to be very, very careful about. So there are a couple of case reports in the medical literature actually, which are allocating certain symptoms to CADASIL in just a child which comes from a CADASIL family. So it's a causality which is being made, which I think is not correct, or it doesn't have any evidence at this point.
There's still a lot of questions on the chat, so maybe I'll slip one in. Has there been any research done with the drug rapamycin? And I believe it's being tested in mice, being done to see if it is beneficial to reduce progression of dementia in CADASIL. Rapamycin, sirolimus?
In longevity, there has, and it can have some we can add it to the list.
It has some severe side effects, though.
True. So we can also complete that part of it. I mean, all of the drugs will have some.
Don't shoot the messenger. I'm just. Another question, more general question about repurposing. What are the initial steps to identifying target drugs for repurposing? Is there a study that has been ongoing or doing that now?
What we want to do is we have at least 2 phenotypes in a dish that we are fairly confident are reproducible and reliable. And so this is really rolling the dice when you're repurposing. You're not necessarily thinking mechanistically, somewhat, but what we are planning on doing is actually FDA-approved drug library screening. In vitro, what's the limitation? There's not really no toxicity there. The repurposing in clinical trials, if we did hypothesis driven, it would be drugs that would have high possibility based on existing targets that are known or diseases in which they've done. And I think the most important thing, if we're going to turn the observational cohorts into clinical trial without concrete evidence, is low toxicity. So low tox is for me really essential in that stratification.
I would say that just like these recent guidelines point out, many of the things that we would normally use to treat small vessel disease or any of the other symptoms in CADASIL, we don't know if they're beneficial, particularly in CADASIL patients. So there's really a huge cohort of drugs we could potentially test for benefit that are already known to be beneficial in people with similar disease that do not have CADASIL.
But I think for us to really hammer that down, we need to figure out the biomarkers, which is research that Fanny's doing and is critical because we need to have endpoints, right? We need to know what it is that we're tracking over time, and can we reverse it? Can we slow it down? There has to be a measurements for us to be able to do this successfully, and we still don't know what that is necessarily.
What you just mentioned is actually really key. I hope that we're entering into an era where we could do rapid clinical testing against biomarkers. So as opposed to waiting 3 years of a large, expensive trial and sort of coarse clinical outcomes, I mean, Julie's imaging data is beautiful, and I think imaging outcomes, but we also have now molecular measures that we think could be surrogate outcomes. And so then we could rapidly move smaller sample sizes, first of all, and then we could rapidly move to the next one. If there's absolutely no impact on the biomarker signatures, move on.
Our time is actually up, believe it or not. Yeah, it has been very good. And again, I want to thank our speakers and great questions coming from patients and the audience, and we'll come back with more answers next year for the next conference. So thank you very much. Oh, and I forgot to remind you of the session at 4 o'clock in this same room this afternoon. The CureCadasil community engagement session that's hosted by the consortium. And then following that, the informal half-an-hour session before dinner. And I had up here the announcement that we're doing fundraising campaign, but that probably goes without saying, you know.
Fundraise for a social worker.
Yes. So again, thank you.